ሰላም ሰላም እንዴት ናችሁ? ዛሬም እንደቀጠለ ነው። ክላስ ነበረ ዛሬ ስለ ኢንዛይም ምናምን የተማርነው። ሪቪው እናደርጋለን ማለት ነው። የተማርነውን ዛሬ ከማንበብ በፊት ክላስ ውስጥ የተማርነውን እና ያልገቡኝን ነገሮች ደግሞ ፊልተር እናደርግና እናነባለን። የትላንትናው ላይ የተወሰነውን ለማየት ሞክሬያለሁ። ጥያቄ ምናምን ደግሞ በኋላ ላይ እንሰራለን። አሁን ጥናት ከመጀመሪያ በፊት ዛሬ የተማርኩትን እንየው ማለት ነው። ዛሬ የተማርነው ስለ ኢንዛይም ነው። Enzymes are proteins and occasionally RNA ይሆናሉ። All enzymes are like by definition they are proteins ግን riboenzyme የሚባል አለ። Sometimes RNA also act as enzymes. That increase the rate of chemical reaction. Enzymes increase the rate of reaction, and there is induced Fate model, and when a substrate touches the active site, the enzyme undergo conformational change to bind the substrate more tightly. Okay, now where is, let me share my screen. Now like delete that guys, so now the whole things. ትላንትና የኤምአይቲ ቪዲዮዎችን ምናምን ለማየት ሞክሬ ነበር። they are pretty በጣም አሪፍ ናቸው። that strategy will be ግን የምጽፈው ነገር አይኖርም። ጀስት ዝም ብዬ ሪቪው ላድርግ። እሺ enzyme ዝም ብላችሁ ስክሪንን ከምታዩት ሳውራ የሆነ ነገር ለፊታችሁ እንትን እንዲል let me share my just screen ላልጽፍ እችላለሁ። you see this, I may not write ብዙ ነገር ግን today we learned about enzyme. All right, what is enzyme? Enzyme is a proteins. Enzymes are proteins, but occasionally RNA known as ribozymes can be also enzymes. Here is an exception. Ribozymes are an RNA and they are also act as enzymes, but enzymes are usually proteins, so they are proteins, and that's and activation energy. Enzymes work by lowering the activation energy required to reach the transition state, allowing reactions to occur faster. Enzymes work by lowering the activation energy required to reach the transition state, allowing reaction to occur faster. Enzymes, how do they increase the rate of chemical reaction? Well, they increase the rate of reactions by lowering the activation energy required to reach the transition state, and allowing reaction to occur faster. And thermodynamically, the enzymes do not change the Gibbs free energy of a reaction. They only change the speed at which the equilibrium is reached. Structural level, there are the levels of organization. Then you have a primary amino acids, sequence of amino acids, and secondary alpha and beta helix, alpha helix and beta sheets. And the tertiary, the functional 3D shape where the active site is formed is in tertiary. And quaternary, multiple polypeptide chains are created, and hemoglobin, here example hemoglobin is an example of quaternary. A specific pocket formed by amino acid that maybe by far apart in the primary sequence, but are brought together by 3 foldings. And this is active site. So each enzymes have this site, and this active site is kind of tertiary structural. And as you know, the primary is just a sequence of amino acid, and kind of its functional unit, but I guess it's a functional unit. So key properties and regulations. Milk conditions, like unlike chemical catalyst, enzymes require specific physiologic conditions. They need specific physiologic conditions. Human enzymes work optimally around 37 degrees centigrade. They denature above 40 degrees, and the pH must be in the range. Okay, so but on the range of specific specific area. For example, pepsin is in the stomach, so works optimally at pH 2, while intestine enzymes work at the pH of 7 to 8, because in the stomach we have HCl, and in intestine mostly they are basics. So basic, and the pH is 7 to 8. The specificity, so absolute specificity and stereospecificity. So when there is okay, let me share the copy, put my notes into the space. So this is my note. So for specificity, da, da, da, da, da, da, da, da. Da, da, da, da, da, da, da, da. So this is specificity. Absolute specificity only acts on one substrate. For example, urease. Urease act only one substrate. And stereospecificity can distinguish between optical isomers. Example, only act on L amino acid, not D amino acids. So can distinguish between optical isomers, only acting on L amino acids, not the amino acids. So this is stereospecificity. And as I know, L amino acids and D amino acids are very different by the last OH orientation. So for example, if it is, this is the structure, and the terminal, this is the terminal side chain. So this is the terminal one. To the terminal, to the last carbon, we have side chain. So if it is on the right, the OH orientation is on the right side, and it will be D. It will call D amino acids. But if it is on the left side, it's called L amino acids. It's kind of distinguishable, L and D amino acids. And but I believe it's different for carbohydrates. Carbohydrates. I will check it out. So enzymes activity is highly regulated by body based on need. Enzymes for glucogenesis are turned on during fasting and off after meal. Okay, so glucogenesis. Let me see what the hell is. So can you? So I want to understand what the hell is glucogenesis, because what is glucogenesis? And we have to know what it is. The light is 19 days, 19 days, 19 days. What is your address? Okay. So how much started you look? Now? Oh, can I understand? Okay, anyways. So what is glyc glucogenesis? And so I have to know this. We have to know this, right? Where was I? And they are. glucogenesis, mild, key. Okay, so the properties. Okay, we covered the properties, and then where was I? Yeah, enzymes regulation. Enzyme activity is highly regulated by the body based on needs. So based on needs, and enzymes are glucogenesis. Enzymes for glucogenesis are turned on. And what are the enzymes for glucogenesis? Because enzymes for glucogenesis. All right, we have to understand that because it's very important. Enzymes are enzymes are uh, activated based on the needs of the body, and glucogenesis is one of the examples. It's activated during fasting, and it's shut off after meal. And so, what are the enzymes like? What are the enzymes, and where is this thing happen? Because I know glucogenesis is happened in liver, but how can we? What's I think this is directly related, controlled by the central nervous system, or or is it? I don't think it is controlled by central nervous system, but it must be controlled by some kind of hormone. All right, because there must be a hormonal activity that's in the higher scheme. Okay, but now we are like zooming in into the biochemistry of the body. Okay, so let's see. And then there is also biological functions. The biological functions, they act as a biological function. They act as a biological function. They act as a biological function like signal transduction, muscle contraction, and jumping. So biological function. Da, da, da, da, da, da, da, da, da. Da, da, da, da, da, da, da, da, da. Dun, dun. Skip one. Oh, Brahman Gannath, Brahman, no, no, no. The Brahman. Then I go back to Zotai. Lotan. Barun per kise, Barun per kise. Activated based on the needs of the body. I think we are smashing on the. Okay. So the biological system. We were ground. Biological function, signal transduction, enzymes like adenylate cyclase convert ATP to CAMP, a secondary messenger in response to hormone. So, what's going on? What's going on? What's going on? What's going on? So. Da, da, da, da, da. Ya Rabbi Batera, Ya Rabbi Batera, Ya Rabbi Batera. So, and like glycogenesis. Glucogenesis was metabolic pathway that allows the body, primarily the liver, to generate glucose from non-carbohydrate source. So this is genesis, okay? Whenever it is glycogenesis, it's a pathway, a metabolic pathway that allows the body, primarily the liver, because the liver is the primary organ that generates glucose. All right. So it's the primary organ that stores glucose and convert them into genetic glucose from non-carbohydrate sources, such as it could be pyruvate. Pyruvate is a three-carbon compound, but it's not necessarily a carbohydrate. All right, and lactate. Lactate is also, what the hell is going on? Like, I believe. Okay, let me check this out. So I believe lactate is carbohydrates, and pyruvate also have three carbon, but I'm not sure. And glycerol is also carbohydrate. I mean, it's glycerol is three carbon I believe it's three carbon carbohydrates. And why it is called carbohydrates? Like, we have to figure out this thing. And so at fasting, certain amino acids, its primary purpose is to maintain blood uh, glucose level during period of fasting or intense exercise when carbohydrates stores are depleted. So when the carbohydrates stores are depleted, so during fasting stage, glycogenesis is necessary because there is lower level of glucose in your body. And what's the implication of that? It's very important. And then uh, so glycogenesis is primarily take place during fasting states, as you know, and while liver glycogen provides the initial buffer for blood glucose between meals, and glycogenesis become the dominant source of glucose as fasting is prolonged. Typically, like beyond 18 ሰዓት በላይ ከሄደ glycogenesis becomes the dominant source of glucose. And liver glycogen ለመጀመሪያ ግን provide the initial buffer for blood glucose between meals. Okay, between meals ላይ ለምሳሌ ቁርስ ሰዓት በልታችሁ ከዛ እስከ ምሳ ሰዓት ያለው ሰዓት ላይ ከዚህ በፊት የነበሩ ግላይኮጂኖችን በሊቨር ዋይል ሊቨር ግላይኮጅን በሊቨር ላይ ያለው ግላይኮጅን as the initial buffer for blood glucose between meals like በጣም ማነስ ያሉ but beyond that ለምሳሌ fasting stage or 18 hour ከሆነ glycogenesis is very important. So the process is highly regulated by hormones like when blood sugar is low, okay, when blood sugar is very low, the hormone glycogen is released. So the blood is low. So ባለፈው ትላንትና ያየነው ነገር ነበረ during fasting stage glucose level low, insulin levels low ይሆናል, but glycogen level is increasing የሚል ነገር አይቻልም። Let me check it out. Let me check it out. Let me check it out. Let me check it out. Let me check it out. Let me check it out. So the insulin level lower, the glucose level lower, but the glycogen level is higher. So when the blood sugar is low, the hormone glycogen is released. And glycogen induces the transcription and translation of specific gene to create the enzyme necessary for glycogenesis. I don't think glycogen is. Oh no, this is not glycogen. This is glucagon. Oh damn. This is my. What the fuck is going on? This is my error, bro. I made an error. This is like በጣም በጣም ቅሌት ውስጥ ነው የገባነው ጋይስ። አልተረዳችሁኝም። what the fuck is going on? እንዴት ነው እንደዚህ አይነት ሹቀት ውስጥ የሚገባው? ማለት ግሉካጎን ግላይኮጅን ብዬ ነው እያየሁት የነበረው አይገርም። ቢካዝ ከዚህ በፊት በማውቀው ስታይል ውስጥ አንዱ ውስጥ ሆኜ ማለት ነው። this is crazy, this is crazy, this is shit, this is shit, this shit is so one. እሺ ግሉካጎንን ነው ግሉኮጅን እያሰብኩት። Okay, let me split a second. So, what's the difference? Like, what's the difference between glucagon, and glucagon and glycogen? Because glycogen is the storage form of the storage glucose, and which is clearly carbohydrate. And during starvation or during fasting, like 3 to 5 day of fasting, then in the blood, glucagon is increased, glucose is decreased, and insulin is also decreased. What is the function of insulin and also glucagon? And all right, so this is very, very important for our understanding. So let's continue. So glucagon is released, and glucagon induces the transcription and translation of specific genes to create the enzymes necessary for glucogenesis. So glucagon is like the enzymes necessary. So because this is vital survival mechanism, the enzymes involved are tightly controlled. If critical enzymes in this or related pathway, like glucagon degradation, is deficient, it can lead to severe hypoglycemia or low blood sugar, as the body is unable to produce glucose to feed the brain and the tissue, because brain is very the primary user of glucose. Okay, በጣም ኢነርጂ ኮንሱም ከሚያደርገው ኦርጋን መካከል አንዱ ነው። but not only the ግን brain can also use ketone body so as an energy source. So ketone body ወደ ሊቨር ይሄዳል። then like መጀመሪያ ከአዲፖዝ ቲሹ fatty acids convert ወደ liver ይሄዳሉ። then the liver convert the ketone body, no, the fat acid into acetyl CoA, then the ketone body liver metabolize ማድረግ ስለማይችል ketone body, it send it into the brain, and the brain goes through its own mechanism, then use it as an ATP. ketone body ይጠቀማል ማለት the brain is like very, very powerful organ. So glucagon is a hormone. So whenever the blood sugar is so blood level is decreasing, in the blood level there is the decrease when there is a decrease in glucose level, and the glucagon level will increase because glucagon is a hormone. So it's induced transcription and translation of specific genes so that they create enzymes necessary for glucagon. So enzymes are proteins, so they are produced in ribosomes. All right, so ribosomes. But why it is RNA ribosome? Let's go back to our process. So glycolysis, there is also glycolysis, breaking down of glycogen back into glucose for quick energy boost. So glucogen. So there, the process are like. Let me write it here. So when they list that train notes. So let me put it down here. The notes. Okay. So these are the notes. Okay. These are our notes, man. My boy, my guy, my guy, my guy, my guy, my guy, my guy. ይዘውረኛል So glycogenesis, glucanogenesis, and glycogenesis. These are very different. Okay, this is glucanogenesis. This is glucanogenesis, and this is so. As you can see, this is glycogen, and then you have here glucanogenesis. So glucanogenesis is creating a new glucose from non-carbs. Okay, this is very important. But glycogenesis is creating glycogen, stored sugar from glucose when the levels are high. Okay, this is very important. ኦኬ ትላንትና በጣም እየተወዛገብኩ የነበረው ዛሬም ቀደም ጀምሮ እየተወዛገብኩ የነበረው is part of my pronunciation. So glycogen and gluconeogenesis. This is gluconeogenesis. This is glycogenesis. So glucanogen is a hormone. So it's induce the creation of glucose from non-carbs. Okay. Whoa, whoa, whoa. ምክንያቱም ግሉኮስ is what? ባለ 6 ካርበን ካርቦሃይድሬት አይደለም? ካርቦሃይድሬት ነው ባለ 6 ካርበን። ሶ እየቀየርን ያለ ነው ነን ካርብስ ናቸው። ዋት አር ነን ካርብስ? ፓይሩቬት። ፓይሩቬት ባለ 3 ካርበን የሆነውን አተም ወደ 6 ካርበን እንቀይረዋለን። ዋት ዘ ሄል ኢዝ ጎይንግ ኦን? እንደዛ ነው እያለ ያለኝ ሳይንሱ። እስኪ let us search. So let us put this way. So glucogenesis is uh, a process where glucose is produced from non-carbohydrates like pyruvates, lactates, and lactates of course, and pyruvate. That's crazy. So how that's possible? Because it's 3 carbon. All right. So pyruvate, if you have pyruvate, you don't have to create glucose. It's easy to create ATP instead of creating glucose into the blood, because that's very nasty. And the science is telling me that we are creating glucose from 3 carbons of other kinds, like from amino acid, from fatty acid, from glycerol. That's crazy. And why it's necessary to create glucose when we can convert it into ATP? Because if you have pyruvate, you have 3 carbon, because pyruvate is an intermediary during glycolysis. All right. So you have 3 carbons, then it's easy to get into Krebs cycle. And what is necessary? So am I correct in my assumption first? Because I may not be correct in my assumptions. We don't know. So but yeah, it is what it is. So glycogen is Glycogen is carbohydrate and it's polysaccharide, and the fuel tank and mostly stored in the liver and muscle. We know glucogen is it's a protein. Okay, glucogen is a protein and peptide hormone. By the way, by the way, ማለት ነው ትላንትና ያገኙት አንድ ሚስጥር ልንገራችሁ። by the way ፕሮቲን that's crazy. This is a heresy. 2001 ላይ ኖቤል ፕራይዝ የበላው ሰው ምን አለ? We don't need we don't need to create peptide bonds, peptide amino acids. We don't need protein አላለም? That's heresy. That's heresy because we know amino acids are the building blocks of protein. But 2021 ኖቤል ፕራይዝ የበላው ሰውዬ ምን አለ መሰላችሁ? We don't need we don't need we don't need like we don't need okay so we don't need proton blood. So we will read about this later. I will read about this later ግን ለጊዜው እንዳለ ይቆይ። So እንዳለ ይቆየን and the function insulin and glucagon. So this insulin ሁለቱ አንታጎኒስት ናቸው. They do the opposite to maintain blood glucose sugar. Glucagon increase እንዲያደርግ የሚያደርገው ነው. So released when the blood glucose is high after eating. So insulin ብዙ ሲኖረን ነው. So after eating fasting ከተባለ ኢንሱሊን የለንም. ኢንሱሊን ይቀንሳል. It acts like key that opens cells to let glucose in. So increased glycolysis አይተናል. Glycolysis is burning sugar for ATP. So insulin የሚያደርገው glycolysis burn ያደርጋቸዋል glucose those sugars burn into. But why man? Like why is it's like for ATP no? Because why calculate for ATP purpose ነው ATP ያስፈልገናል። ግሉኮስ የማት ግሉኮስ ስላለ ለATP ነው የሚፈልገው because we need ATP. And increase glycogenesis making glycogen to store energy ደግሞ እንዲሆን increase ያደርጋል. So burn ይደረጋል ለሌላው or ደግሞ and decrease blood sugar level. So blood sugar level ደግሞ decrease ያደርጋል ማለት ነው. But what is the reason behind the blood glucose level lower ማድረጋችን? What is the rationale behind it? Because what is necessary? Blood glucose በጣም accumulate ቢሆንስ? What the fuck? Because it's homeostasis አይደለም? Because it's a certain range of blood sugar in our blood is መሆን ያለበት. So out of that range ሲሆን it's out of nature. So decrease እንዲያደርግ ያደርጋል to maintain homeostasis. That's my speculation, but there must be other reasons. So glucagon is fasting state. ለምሳሌ ምግብ ከበላችሁ በኋላ or after meals በምግብ መካከል የሚኖር ምናምን ነው. So increasing glucogenolysis breaks down glycogen. Glucogenesis make new glucose from amino acid in the fats. So increase glycogenolysis breaking down of glycogen and increase blood sugar level ደግሞ increase ያደርጋል. So let's say starvation ከ3 እስከ 7 ቀን የሚቆይ 5 ቀን ምናምን ምግብ ያለ ምግብ ከሆናችሁ immediate fasting ለምሳሌ blood sugar drops ያደርጋል. So insulin secretion stop because increase insulin secretion ይቆማል። ምክንያቱም insulin exists when there is glucose ሲኖር. So insulin የሚወጉት ለምንድን ነው ከተባላችሁ በቃ ምግብ በልተው ያ ግሉኮስ የትም አይሄድም። ሶ ያ ያ ሹገር ባይኖር ኖሮ ሹገሩ ስኳር አምናት ጊቪንግ ኤኒ አድቫይስ አስ ስቱደንት ዲስ ኢዝ ማይ ስፔኩሌሽን ገና ማስተር አላደረኩትም ሳብጀክቱን ግን ሲዋጉ ማይ ስፔኩሌሽን ኢዝ ኢንሱሊን አይደለም የሚዋጉት which is a hormone which aid them to utilize those glycogen. So so so whoa, whoa, whoa, whoa, whoa. So whoa, whoa, whoa, whoa. So whoa, whoa, whoa, whoa, whoa, whoa. All right, so okay. Okay, የሆነ ነገር እያየሁ ነበር አሁን እና let's go back. So insulin lowers and so the first 24 hour travels to the liver and force it to break down glycogen. ግሉካጎን ምንድነው የሚያደርገው? ግሉካጎን ኢንሱሊን ሎወር ነው የሚያደርገው፣ ግሉካጎን ደግሞ ኢንክሪዝ ነው የሚያደርገው። ሶ ምግብ መብላት አቆምን አይደለም። ሶ ኢሚዲየት ፋስቲንግ ከሆነ ብለድ ሹገር ድሮፕ ያደርጋል፣ ኢንሱሊን ሴክሬሽን ይቆማል፣ የግሉካጎን ስክሬት እየተደረገ ይሄዳል። ሴክሬሽን በደንብ then the first 20 hours መጀመሪያው ላይ ምን ይሆናል? ግሉካጎን ወደ ሊቨር ይሄድና it forces down to ግላይኮጂንን ብሬክዳውን እንዲደረግ ያዛል ማለት ያዘዋል። ሶ ባይ ኤንድ ኦፍ 24 hours your liver glycogen stores almost completely empty ይሆናል ማለት ሶ ስቶር የተደረገው ግላይኮጅን ሊቨር ላይ ያለው ግሉኮስ በግላይኮጅን ፎርም ስቶር የተደረገው ዩዝ አፕ ይደረጋል፣ ጥቅም ላይ ይውላል። ሶ ግን ከ3 እስከ 7 ቀን ያለው ሲንስ ግላይኮጅን ኢዝ ጎን ሶ ግሉካጎን stays high to tell the body we need fuel። ስለዚህ glycogenesis the body starts breaking down muscles protein to get amino acid and fat tissue to create new glucose in the liver. So fats to create new glucose in the liver. So liver ላይ ነው ግሉኮስ የሚደረገው። So ketogenesis since glucose is scarce the body begins converting fatty acids into ketone bodies። ወደ ኬቶን ቦዲ ይቀየራል። የት ነው የሚቀየረው? አሁንም ጭንቅላትን ለመመገብ ማለት ነው። አሁን እዚህ ጋር ምስሉ ላይ ያየ ነው እዚህ ጋር ያየ ነው ኬቶን ቦዲ ለጭንቅላት ነው የሚላከው፣ ለሌላ አካል አይላክም። እዚህ ጋር ባየሁት መሰረት መስል ላይ ምናምን ይደረጋል። ከመስል ላይ ፕሮቲኑ ወደ የት ይደረጋል? ወደ ግሉኮስ but the fatty acids is converted to ketone body. So ketogenesis ይባላል ማለት ነው። Since glucose is scarce, the body begins converting fatty acids into ketone body to feed the brain and usually act as alternative ATP source to glucose. So insulin lower blood sugars by storing it as a glycogen or fat, and glucagon raises blood sugar by releasing it from glycogen and making it new. All right, so that is very important. ከዛ ውጪ ማየት የምፈልገው ነገር so so ግሉኮስ ከሌለ our body may die off. So ሁሉም የሰውነታችን ክፍሎች ግሉኮስን ማምረት አይችሉም። ኦኬ ለምሳሌ ሬድ ብለድ ሴል ማይቶኮንድሪያ የላቸው፣ ግሉኮስ አይመረትም። ባይ ዘ ወይ ግሉኮስ ሲጀምር የሚመረተው በምንድን ነው? ፈርስት ኦፍ this is ስፓይራል እያደረገ ነው የሚለው። ኤስ ካርቦሃይድሬት ከምግብ ነው የምታገኘው። ግሉኮስ ምግብ ውስጥ የምታገኘው ኢነርጂ ነው። ኦኬ ከምግብ ነው የምታገኘው። አሚን ከምግብ ነው የምታገኘው። ካልሆነ ግን ያን ሜቴጌዝ የሚያደርጉትን ነገሮች ይሰራልሃል በመቀየር። ለምሳሌ ግላይሰሮል ወደ ግሉኮስ ሊቀየር ይችላል። 3 ካርበን ኮምፓውንድ ነው ግላይሰሮል። አይ ቢሊቭ ኢት ኢዝ 3 ካርበን ኮምፓውንድ አይደለም ግላይሰሮል። It's fatty acid. Glycerol is 3 carbon compound. So glycerol, this is glycerol, okay? What formula is this is this is glycerol. This is glycerol. Okay, this is glycerol. This is glycerol. This is glycerol. So እነዚህ ኮምፓውንዶች they will be converted into glucose. Like ለምሳሌ one of my question is so again like one of my question is እዚህ ጋር ያለው ግሉኮስ አለ። there is here we have a glycerol, so which is C3 all right, H8 it's H8 1 2 3 4 5 6 okay, H6O3. This compound is converted into glucose in the liver. My question is this: why do we need glucose in the first place? Because my question is this: is it necessary to convert glucose, this glycerol into glucose for the use? Because in glycolysis, the end product of glucose is ATP. So why don't we just change this glycerol into ATP? So this is one of the question. So this question have one assumption, which is glucose is only used for ATP production. ATP production. So is it true? Or else glucose is used for other purposes as well. All right, this is my first question. The second question is how glucose is produced in our body. Is it directly getted from the food we eat, or is it there is a process where glucose is produced? Like, because how glucose is produced in the body. I know glucose is stored in the liver, but how glucose is produced and where it is produced. So I want to know about this before we close up. And pyruvate is also the Krebs glycolysis. Gly- So there's so like ለምሳሌ brain blood barrier, while brain can eventually use ketone as so it cannot use fatty acid usually effectively because fat molecules are too large to cross blood-brain barrier rapidly. Further, even in deep starvation, the brain still require small baseline amount of glucose. So while brain eventually use ketone ሊጠቀም ይችላል ነገር ግን so often turning pyruvate into glucose cost energy. Yes, of course it costs energy. ሁለት ATP ይፈልጋል። Oh, no. This is reversible reaction. This reversible reaction. Turning pyruvate reverse with a glucose is glycogen cost energy, where turning pyruvate into ATP, Krebs cycles, energy generates energy. So why the body spend energy to build glucose when it's starving? Like so, this is very okay. So yeah, my question was very necessary. And the liver perform glucogenesis not for itself, but to keep the brain and red blood cell alive ለማድረግ ነው ማለት ነው። So why not just turn pyruvate into ATP? ፓይሩቬቱን ለምን ወደ ATP አንቀይረውም? Why not just turn pyruvate into ATP? So the liver does make ATP, but it makes it from fatty acid oxidation. During starvation, the liver is, I think, yeah, this ATP produce ያደርጋል, but from fatty acid ነው። እዚህ ጋር clearly image ገልጾታል። It then uses the ATP to pay the expensive cost of converting pyruvate to glycolysis. The liver burns fat, ATP source, to build glucose, fuel source. So the liver burns fat, ATP source, to build glucose fuel source so that it can ship the glucose out into the blood from the brain and the RBC. So this is the rationale. So the liver burns fat ATP to build glucose so that it can ship the glucose. Oh, now I get it. So the liver actually produce here this here image, this image line. You can see this reaction now. Ziga, you can see it. Ziga, this part here is Ziga fat. Ziga fat is converted into Ziga ATP. So this is ATP or ATP man ይቀየራል. Right? Then what ATP ይቀየራል. So this ATP is used so that glucose እዚህ ጋር ያለው glucose so that ወደ brain ምናምን እንዲሄድ ያደርገዋል ማለት ነው። ወይም ወደ RBC እንዲሄድ ግሉኮስ ወደ RBC እና ወደ ብሬን እንዲሄድ ያግዘናል ማለት ነው። So this is the rationale. አንቺ ቅመም፣ አንቺ የሰው ቅመም፣ አንቺ የሰው ቅመም፣ አንቺ የሰው ቅመም፣ አንቺ የሰው ቅመም በፋቲ አሲድ ኦክሲዴሽን ነው። So ዛሬ ማየው ምንድን ነው? ካርቦ So ዛሬ የምናየው I found a great YouTube channel the whole የሚያሳይ ኢንዛይምስ ኤንድ ሌሎች ሪአክሽኖችን እነሱን ያለው። ከዛ ውጭ ግን I want to revise this oxidation, dehydrogenation, covalent bonding ምናምን ኪክ መደረግ አለባቸው። ደን ዛሬ እነሱን ያለው ነገ ከሰዓት ክላስ የለንም። ከሰዓት ጀምሮ አርብ የለም፣ ቅዳሜ የለም፣ እሁድ የለም። ሰው አርብ፣ ቅዳሜ፣ እሁድ we will do other subjects guys. So lactate, lactate. So recycle the recycle mentioned lactate. This is a perfect example why glycogenesis is necessary. Muscle exercises and produces lactate anaerobic. Blood lactate travels to the liver. Liver converts lactate, pyruvate, glucose. Glucose goes back to the muscle to be used again. La liver converts lactate um, into pyruvate. Mm, Lactate converts into pyruvate and finally into glucose. And the blood lactate travels to the liver. Lactate travels to the liver. Muscle exercise and produce lactate. Glucose goes back to the muscle to be used again. If the liver just burned that pyruvate for ATP, the glucose supply would vanish. So, and the muscle and the brain would run out of fuel. So, the liver just burned the pyruvate for just ATP, the glucose supply would vanish, and the muscle and the brain would. So, this is kind of protective. So that's great. That's great. That's great. That's great. So why is glucogenesis necessary if it is energetically expensive? Glucogenesis is necessary because certain tissues, specifically red blood cells, which lack mitochondria, and the brain, due to blood-brain barrier, are obligate glucose consumers, and they cannot oxidize fatty acids for energy. Therefore, the liver must expend energy to synthesize glucose from precursors like pyruvate, lactate to maintain blood glucose level and prevent hypoglycemia. So ሃይፖግላይሲሚያን ለመከላከል ይጠቀምበታል። ከዛ ውጭ ግን ምን ያደርጋል ማለት ነው? ፋቲ አሲድን እንደ ኤቲፒ ይጠቀምበታል። ሶ ዳት እዚህ ጋር የተመረተው ግሉኮስ እንድትሄድ ያግዛለች ማለት ነው። አገኘህ አገኘሽ አግኝተኛ ቴንኪው አመሰግናለሁ። ሳይንሱ እንደዚህ ነው። ዊጌት እሺ ቀጣይ so oxidation fatty acid and the ketone bodies during fasting muscles oxidize fatty acid released from adipose tissue and ketone bodies produced by the liver during exercise muscle can also use its own glycogen store as well as glucose fatty acids and the ketone bodies from the blood. So this is very very essential. So muscle decrease its use of ketone bodies and oxidized fatty acid as its primary energy source. Ketone body መጠቀም ያቆምና because what is the rationale እዚህ ጋር? So the brain takes and oxidize ketone bodies to drive energy. Consequently, the brain decreases its use of glucose, although glucose is still a major fuel for the brain. Oh. And liver glucogenesis decreases, and muscle protein is spread less. But the question is, so now the second question is this. So we got the rationale behind why glucose is essential, because glucose is the production of glucose is essential for certain bodies because they only use glucose as their fuel. But how they use it after glucose is get into some of the tissues, for example, in red blood cell. Red blood cell is also connective tissue, right? Red blood cell. So once, because red blood cell doesn't have mitochondria, and we know oxidative phosphorylation cannot be happened in the red blood cell, because even if we have a high amount of oxygen, we don't have mitochondria, and we don't so glycolysis cannot be happening. So what is the use of the advantage or the process of glycolysis? Glucose, glucose in the brain, because how brain use glucose? How brain, because we know brain use glucose, and we have said that, but how it use it? Is it a glycolysis, or there are other mechanisms as well? Alrighty. Alright. በዝምታ መሃል ዝምታዬን ወሰድኳት። ዝም ብዬ አልቀርም እናገራለሁ ቃሌን አዘምራለሁ። ዝም ብዬ አልቀርም። እንመለስ። የዛሬውን ሪቪው አደርጋለሁ ብዬ ሌላ ዓለም ውስጥ ገባሁ። So metabolic origin, lactate often comes from muscle activity, anaerobic metabolism, pyruvate, the end result of glycolysis that can be recycled. Glycerol released when fat body fat. Triglycerols is broken down for energy. Triglycerol breakdowns in glycerol and fatty acid, because they're the components. Amino acid derived from breakdown of proteins. So yo. So creating glycogen stores sugar from glucose. Glycolysis, breaking down of stored glycogen back into glucose for quick energy boost, because body use glycogen by the way red blood cell ግሉኮስን ወስዶ ወደ ላክቴት አሲድ ላክቴትን ነው የሚጠቀመው አይ ቢሊቭ፣ ቢካዝ እዚህ ጋር አለ ምስሉ ላይ ላክቴትን ነው የሚወስደው። ግን ላክቴት ዋት ኢዝ ላክቴት ባይ ዘ ወይ ዋት ኢዝ ላክቴት የሚለውንም እናያለን። ሶ ሌትስ ጎ ባክ ቱ አወር ስተዲ። ብዙ ወደኋላ እንዳንል እንግፋና ወደፊት። So there are clinical cases as well. So እዚህ ጋር አየን። So signal transduction enzymes like adenylate cyclase convert ATP to cAMP, secondary messenger, in response to hormones like epinephrine. Enzymes like adenylate cyclase convert ATP to cAMP, secondary messenger, in response to hormones. Signal transduction. So signal transduction አይተናል እንዴ? ከዚህ በፊት አላየንም፣ እናየዋለን በሌላ ጊዜ። ግን this is also adenylate cyclase የሚባል የኢምፖርታንት ፈንክሽን እንዳለው እናያለን። So involving glycolysis, glucogenesis and recycling ላይ also very important ብሎናል። muscle contraction, enzymes like myosin kinase facilitate movement, and iron pumping enzymes like sodium potassium ATPase. This is pumper maintain cellular gradients. So clinical and diagnosis importance. Enzymes serve as biomarkers for identifying organ damage. So myocardial infarction, heart attack, elevated level of CKMB, creatine kinase MB in the blood indicates heart muscle damage. So liver function test elevated ALT and AST indicate liver injury or inflammation. So inborn error of metabolism, PKU or deficiency in phenylalanine hydroxylase managed by low diet and tyrosine supplementation ይሆናል ማለት ነው። ኢንቦርን ኢረርስ ለምሳሌ ህፃናት ላይ there can be deficiency in so phenylalanine hydroxylase ኢንዛይም ካለ የዚህ እጥረት ካለ የሜንታል ሪታርዴሽን ሊገጥማቸው ይችላል ማለት ነው። ቢካዝ ለምን ከተባለ this is like this is phenylalanine is very important amino acid, very important amino acid no. So this is amino acid which you might get it from food. So essential ስለሆነ ከምግብ ልታገኙ actually ከምግብ ልታገኙት አትችሉም። Is it? I believe it's essential, but is it that much essential protein እንደሆነ አውቃለሁ። Let me search this. ረስቼዋለሁ or አልተማርኩትም። So it's essential amino acid ነው። So the formulaውን ማወቅ ከፈለጋችሁ እሺ ላስቀምጥላችሁ እዚህ ጋር Google is very powerful. አሁን እዚህ ጋር የጻፈውን ላሳያችሁ ከጉግል ነው የወሰድኩት። Google is very powerful. So screenshot ባረግ ይሻላል የጉግሉ መልስ። Guys, like people's like it doesn't matter, it is okay, I know myself, you know your name, do you know my name? No, you know my name. No, you know my name. Alrighty. Okay, so እዚች ጋር ምን እናደርጋለን መሰለሽ? እቺን እንወስዳለን። So this is very essential amino acid. Okay, if it is essential ከሆነ ምንድን ነው? ምን እያለን ነው? ከምግብ ነው የምታገኘው እያለን። So crucial for protein synthesis and neurotransmitter production like dopamine, norepinephrine, and found in high protein foods like meat, dairy, and eggs, is converted into tyrosine in the body. Tyrosine is like non-essential now, so it is used for potential benefit in treating depression and chronic pain and the skin conditions. So it's dangerous individual with PKU. So the role is its essential amino acid and the neurotransmitter cursor and protein building block and the metabolism ላይ ያስፈልገናል ማለት so especially ወደ ታይሮሲን ይቀየራል። so who is ታይሮሲን ምን አይነት አሚኖ አሲድ ነው? አሁን እቺኛውን አሚኖ አሲድ አያችሁ? በቃ አሚኖ አሲድ ስትባል C9H11 NO2 ምን አይነት ስትራክቸር ነው ያላት? ባይዘዌችን ስለ ፌናይሊን አንድ ነገር ልንገራችሁና አልነለም። ፌናይሊን is kind of ሃይድሮፎቢክ ሳይድ ቼን ያለው ሃይድሮፎቢክ ሳይድ ቼን ያለው እንትን ነው ማለት ላሳያችሁ። ስሪዲው ሲሰራ በዚህ ነው የሚሰራው። this is its structure ታምራት ነው እኮ የሚያሳየን እዚህ ጋር። ታምራት እኮ ነው የሚያሳየን እዚህ ጋር። በቃ ታምራት እያየን ነው። this is phobic now, so this is the side chain, this is the side chain, this is the side chain, this is the side, this is the side, This is the side chain. The side chain. What the hell? Da da da da da da. Da da da da da da. Da da da da da. What? አይሰራም እንዴ? እንደዚህ ብዬ ልሰራው ነበር። What? ቢገጥም ባይገጥም እኛ ምን አገባን? ሲቹዌሽን እስኪ እሺ so this is the side chain እና ይሄ this is hydrophobic ነው. So this is NH2O, this is carboxylic አለልን NH2 አለ. This is the side chain, this is the R part እና this is kind of ring structure ነው. አንድ ሁለት ሶስት አራት አምስት ስድስት ካርበን አለ። በዚህ በኩል አንድ ሁለት ሶስት አራት ካርበን አለ። አንድ ላይ ሲደምራቸው ስንት ይሆናል? 9 ካርበን ይሆናል ማለት so where is the this is 1 carbon እዚህ ጋር አንድ ካርበን አለ። እዚህ ጋር ሁለት ካርበን አለ። this is 3 carbon, this 3 okay so አንድ ሁለት ሶስት what is this? Actually this is not carbon and 9 carbon alan. This one is hydrophobic and I believe it is charged. And tyrosine is also convert into tyrosine. So tyrosine እንመልከታት tyrosine is non-essential. It is not essential. Tyrosine is so ወደ ታይሮሲን ይቀየራል። ታይሮሲን ሰፕሊሜንቴሽን ያስፈልጋል። ባይ ዘ ዌይ ታይሮሲን ሰፕሊሜንቴሽን የሚያስፈልግ ከሆነ it must be some kind of ከምግብ የምታገኙ or what ሰማይ ነው የሚሆነው እርግጠኛ። ታይሮሲን ፎርሙላን እንመልከት መጀመሪያ እንመልከት። This is tyrosine. So this is the side chain. This is the side chain, and I don't think this is problematic side chain. እንደምታዩት ያው ጽድት ያለች side chain ናት። So this OH group is very great እንትን because hydrophobic መሆን አይችልም ይሄ ነገር። this one ታዲያ እንዴት ነው hydrophobic ውስጥ ያስገባሁት? ነን ኢሰንሻል ነው። ኦኬ ኢሰንሻል አይደለም፣ ነን ኢሰንሻል ነው ቢካዝ ባዲው ነው ኮንቨርት የሚያደርገው ከፌናይላይሊን ሶ ለስትረስ ለአለርትነስ ምናምን የሚሄድ ነው። and serve as a critical building block for protein and key brain chemicals including adrenaline and norepinephrine ለእነዚህ የምትሆን ናት ማለት ነች። very important የሆነች ኢለመንቶሲስ ናት ማለት ነው። therefore I am tyrosine. So alertness, dopamine and adrenaline and norepinephrine and dopamine የሚጠቀም ኬሚካል ኢሰንሻል ኬሚካል ናት for this ለእነዚህ sure so thyroid and skin house and for thyroid hormone and thyroxine and melanin the pigment responsible for skin ለሙድ ለምናምን ትጠቅማለች። አይ ቲንክ እየጣፈጠኝ ነው ይሄ ትምህርት። የሆነ ሹገር እያለኝ ነው። ሹገር ሲጀምር አታካብሩ ላይክ what I want to do ለምን ፐብሊክ እንደማደርገው ልንገራችሁ። ሶ ማይ ፈርስት ኤግዛም አይ ፌልድ አ ላት እና ከአሁን በኋላ ግን አይ ወንት ፌል ከአሁን በኋላ ቢካዝ አም ጎና ወርክ ፐብሊክሊ የማደርገው ዲስ ፕሮሰስ ኢዝ አይ ዊል ናት ፌል አጌን በወርክም እነሳለሁ ግን ዜር ኢዝ ኖ ፕሮሚስ ፎር ፌሊየር የምፈልጌ አልወደኩም ለነገሩ አልወደኩም So why turn glycerol into glucose? Why not just turn it into ATP? Is it necessary to convert glycerol into glucose? And can't we just change it to ATP? Is glucose only used for ATP? Yes, the liver can burn glycerol directly for ATP. So is it necessary to convert glycerol into glucose? Can't we just change it into, yeah. So glycerol point, glycerol enters metabolic pathway at the midpoint of glycolysis, like this glycolysis. ምንድን ነበረ ቀደም ያየሁት አዮኖ ምንድን ነበር ያለችኝ ያች ፕሮፌሰር አዮኖፎን ነው ሊኮን ሌክሲዮን ሌክሲኮን lexicona, lexicona, lexicona, MIT biological chemistry, lexicona, lexicona, lexicona, lexicona, lexicona, lexicona. ሰላሳ ስምንት ፔጅ ነው ኦኬ I find it finally. ወየው ጉዴ እግዚአብሔር ይስጣችሁ። ኤምአይቲ ኤምአይቲ ኤምአይቲ ምን እንደሆነ ታውቃላችሁ? ኤምአይቲ ማለት መቀሌ ኢንስቲትዩት ኦፍ ቴክኖሎጂ ቲች ምንድን ነው እየሰራችሁ ያላችሁ? ስዕሉን አትሰጡኝም ነበር? ዋት ኢዝ ዘ ሆል ኦፍ ጎይንግ ኦን? እስኪ አሁን እዚህ ጋር ሌክሲኮኗ ሰጥታኛለች። ሌክሲኮኗ የቱ ጋር ማየት እንችላለን? ግላይኮላይሲስ የቱ ጋር ነው ያለው? ግላይኮላይሲስ ወይ ግላይኮላይሲስ ቢዮቲን። ዛሬ ስለ ቢዮቲን እያወራ ነበረ አስተማሪያችን፣ ስለ ቢዮቲን እያወራ ነበረ። ቴቴማሬ ፓራፓራራ ቴማሬ Oh mama, oh mama, what's going on now? Guys, እዚህ ጋር guys ሼር ላድርጋችሁ። ለሚ guys የሆነች ዶክተር አለች እንደዚህ ያለች ነው። This will be our first reaction video, okay? How we react? Let me start. Let me start. Tettamura bitti mare parateram tarimare. Arantarosturum barendarendarandaristaru. Oh mama. Boom. Okay, let me share it. Let me share it. ግንባር ነው ያስመሽ what the fuck አራዳ እንደዚህ ነው የሚያደርገው so let's react to I'm kind of bored so let's watch a video together እና like ዶክናት so እየተመረገች ምናምን ነው የሚመስለው so let's first see Ooh, wow, wow, wow. This is crazy. Ooh.
Hi guys, እንዴት ናችሁ? Welcome back to my YouTube channel. It's Rutina.
Yeah.
እንደምታዩኝ አስታውቃለሁ ከፀጉር ቤት ነው የመጣሁት። ተሰፍቼ ማለት ነው my human hair. I don't know if you guys remember this human hair. It was a brown. በጣም የሚያምር ነበር. I hope I'm not gonna regret it because በጣም የሚያምር ከለር ነበር። ግን በቃ ፀጉሬ ጋር ሄድ ስላለኝ ጥቁር ፓርት እንትን እንዲህ ብዬ ሙሉ ለሙሉ ጥቁር አደረገው። I didn't want it to be like this, but እስቲ ሲታጥብ ይለቃል ብሏል። እንግዲህ we'll see about that. እንደዚህ ፏፏ እያልን ያለ ነው ለመሰላችሁ tomorrow's the day, the big day የምንመረቅበት. We are finally gonna graduate tomorrow. የት ነው የሚሆነው? ሀይሌ ግራንድ ሆቴል
የት ዩኒቨርሲቲ
ነው የሚሆነው። ፏ ማለት አለብን። I can't wait to show you guys my dress. Oh my god, I don't know you guys, I don't know. እና ፀጉሬም አይ ቲንክ አሪፍ ነው ከሱ ጋር ሄዳል። yellow, my dress is yellow እና ጠቅልዬ ለማሳደር ነበር ሀሳቤ መጀመሪያ ላይ፣ ግን እንደማየው ከሆነ የሚበላሽ አይነት ፀጉር አይደለም። እንደሰራኝም ሰው ወደ ላይ ዝም ብለሽ አስቀምጪ ነበር ያለኝ። so I'm gonna do that because ብትን ሲል ነው የሚያምረው እንደዚህ ሆነ። ዛሬ አርብ ነው ፕሮግራሙ፣ ነገ ነው እንዳልኳችሁ ቅዳሜ ቀን ማለት ነው። Hey guys, finally graduation ልብሳችን ምናምን ተቀብለናል። we paid for it like ከውስጥ ቲያራ እንደዚህ አድርጌ ለጥፌያለሁ። በደንብ ቆይ በፕላስተር ለጥፈዋለሁ ቁጭ እንዲል ማለት ነው። አይደው ነው ከፊት እንዲህ ምናምን እዚህ ድረስ እንዲመጣ አልፈልግም። ዝም ብዬ ከኋላ አድርጌ ቁጭ ለማድረግ ነው ሀሳቤ። ፊት ለፊቷ ፀጉር የሆነ ከርድ የሆነ ከዚህ ጀምሮ ብትሆን አሪፍ ነበር። እስቲ ቆይ ጠቅልዬ አሳድራታለሁ ቲያራውን ፀጉር ውስጥ እንሰካዋለን ማለት ነው። ቆይ ነገ በጠዋት ነው ግራጁዌሽኑ፣ ሁለት early በጣም early, so I had to get my hair done and also I'm gonna do my own makeup በጠዋት so yeah ለይተን ፏፏ እንላለን። እና ቪዲዮ ዛሬ መጀመሪያ አሪፍ ነው ምክንያቱም ነገ በስርዓት እንደዚህ ራሱ አላወራችሁም ጥርፊያ ላይ ስለመሆን ማለት ነው። ቀሚሴ ራሱ ትንሽ ትግል የምትፈልግ ቀሚስ ናት። ከኋላ ገመድ ኮርሰት ነገር ውስጥ ይታሰር ምናምን and so my dress was ረዥም ቀሚስ ነበር። አሳያችኋለሁ ነገር ማይንስ ረዥም ነበር and then I'm going to keep my cap for the rest of this video ብቻ ረዥም ነበር and then my friends were like ጋውን ረዥም ስለሆነ ቀሚስ ማብሮ ረዥም ሲሆን it's kind of weird ነገር and then I was like ከዛ ደበረኝ። በቃ ኮተት የሚሆን መሰለኝ ነው ቀሚሱም ሲረዝም ከጋውኑ ደግሞ ዋናው አጭር ይሆን ብዬ ነበር። I kind of regret it now ረዥም ቢሆን በጣም ረዥም ቢሆን አጓጉ ነገር ቢሆን አሁን ግን it's very short like እዚህ ጋር ሆና ግን እንደዛ ሆና ግን it's really cute አሳያችኋለሁ ነገር anyways ፀጉሬ ቲያራ በደንብ ነው የያዘው እየተነቀነቀ የለም so body care ምናምን ከዛ ላይ ደግሞ I was doing that to take care of my skin then what should we do በቃ እስቲ ጁስ ምናምን እየጠጣን ሰሞኑን እሱ ነው ሳደርግ የነበረው I was drinking juice I love my carrot juice በጣም ወደው and የተለያየ fruits ምናምን እየጠጣሁ ነበር maybe እሱ ይጠቅማል and I need to rest አሁንም ቢሆን በቃ ይሄን ቪዲዮ ልስራላችሁ ብዬ ነው ሰው ላሳያችሁ ብዬ ነው ትክክል አድርገን ሳይን በልቼ አልጋዬ ውስጥ ላጥ ፊልም እያየሁ I have to rest. This is the hair. It's orange juice. I'm in love. We're gonna enjoy the rest of the day. That's what I know. I'm gonna cry. I'm not gonna cry, but I'm gonna shed some tears. Honestly, you guys, we've been waiting for this day for like for 7+ years. Like, this is crazy. And yeah, I've lived 7 times up. It's just not. It was full of hardship, you guys. Full of emotions. Full of bad memories, good memories. Medical school is not easy. It's so hard. But I'm so glad I went through it and I achieved my goals on it. Because I didn't thought like I was like, should I quit? Like let me see, but it would like why? It was always why, and I almost got into depression now, man. I never thought in my life I can't believe it. I'm so happy, so happy that I didn't quit and that I made it through. I'm so proud of myself right now. I am so proud of myself. Like medical school, I wanted to pursue for my future. So like I also did social media. It was not easy. Like I'm not sometimes on social media. Life is good and easy. It was not. it was not እና ብዙ ሰው like ጓደኞቼ አብረኝ የሚማሩትን የሚገርማቸው like በምን ሰዓት ነው ሶሻል ሚዲያ ምናምን ትራሱ it was I don't know how I did it but I'm so glad I did. I feel like ትንሿ ሩት would be so proud. Oh my god ዛሬ አልቀሳለሁ ዛሬ so my family አብረን ነው የምንሄደው. my two sisters አሉ they're going to be proud. They're proud already. But my family, my parents and sisters ሄደን በጠዋት we're gonna ቃል ማለም ገባለን ምናምን እዚህ ከቻልኩ ቀስላቸው the whole thing ስኪያብን ከሆነ I'll try to yeah I'm gonna do that. It's a vlog so we're not gonna ruin it. anyways ፊት ትንሽ አባብጧል አውቃለሁ ከእንቅልፍ ስለተነሳሁ ነው። አሁን መዘጋጀት እንጀምር and ፀጉሬ ልብሴን እስክታዩ ቸኩያለሁ ማለት ትንሽ አጥሮ እንዳልኳችሁ I don't like that part but እስቲ we'll see. ሲጀመር ጋውን የምናደርገው the whole time so I don't think የሚታይ የሚያል እንደዚህ ነው። tripod የተበላሽቶ በዚህ አስተክፍኝ ያለው so I was going to do my makeup and then I remembered ካርቴን ዛሬ ነው የምሰጠው። ካርቴን ምንም አሳያችኋለሁ በሌላ ቪዲዮ የዲክስ ባለ ኔክስት ዊክ ነው የዛሬ ሳምንት ነው እና for my friends አጻፍኩና ቶሎ ቶሎ እንጻፍላቸውና በሰበቡ ዛሬ እሰጣቸዋለሁ ሁሉንም ዛሬ ስለማገኝ ማለት ነው። ከዛ ፎቶ ውስጥ ከነ ባለቤቱ ከነ ምናምን የሚለው ፓርት የጻፉት and then the outer part ናቸው ማለት ነው። ቶሎ ቶሎ እናድርግና we're gonna start on she has my setup by the way. This is my makeup piece. My makeup spot. Makeup ቶሎ ቶሎ መቀባት ስላለብኝ I'm not gonna show you guys እያንዳንዱ ውስጥ ተቀምጬ ነበር ከሳንት ወደ Dream about us together again. What I want us together again, baby. I know we'll be together. I am so glad and happy I can do my own makeup. ምክንያቱም በዚህ ለሊት ሜካፕ አርቲስት ዛሬ ምን እንደምናደርግ ነገራችሁ። so the plan is ጠዋት 2 ሰዓት ፕሮግራሙ ይጀመራል and then እስከ 6 ነው እንቆይ ይመስለኛል። ከዛ ሀይሌ ግራንድ እናልኳቸው አይደል እዛው ምሳ ከፋሚሊ ጋር እንበላለን ሰብሰብ ብለን and then ቅርብም ስለሆነ አሪፍ ነው። ከዛ ወደ ቤት መጥቼ ደግሞ ለብቻችን our batch skylight ቡክ አድርገናል እራት ለመብላት ማለት ነው። አብረን የምናየው ይሆናል። Okay guys, this is the dress. Now ፀጉሬንና ጆለሪ ምናምን ቶሎ ቶሎ እናስተካክል። the struggle ከማም ጋር ያየ ነው ማለት ነው። ስትቋጥርልኝ ከኋላ POV bro I can't tell where your hair ends and mine begins. If I had a body tattoos I would think that it's yours, yeah. If I move too quick that's you I would think it's my affection. In this cold city close enough who could tell me to touch between us. Little me to the apartment one day I'm ready to heart it. It's the stairs but we caught it. Rise to that feeling to fire it. I know when you see just one you can see us too.
She already made it. I get it. Nice sunshine maybe. Bo Daryan'ın right. İstanbul'lara gelebilir. Yetmez. Yatırımları. Congratulations. Hayat. Oh. Hayat Medical College. Hayatoች አሪፍ ናቸው። So waiting area
ሰው እስካሁን የጠፋሽ ካላችሁ ቪዲዮ ራሱ እየቀረጽ ካለ የጠፋሽ ካላችሁ ሪቫን ለካ መምጣት ነበረበት አላወኩም አልሰማሁም። እንዲህ ከቤት አምጦልኝ ቤት ትንሽ ቤት ነው ተረጋግቻለሁ። ዶክተሮች የት እንደምታገኙ ራሳችሁ ታውቃላችሁ። From the graduating class.
For your exceptional mentorship, support, and commitment to train future physicians, thank you.
So our favorite department, surgical department, no, of course. Not the favorite senior set up from surgery department, Dr. Mikas. Can alternative መሰለኝ መጣ ይመስለኝ we are. So this is the gift. Gift card orders ሆነ የተደረደረ ነው።
ትንሽ ተማሪዎች ነው። Like, just a minute. For 5 years? Right. What is the gift family? Well, we will persist. We will persist. I will persist. I will persist. This is the roots. This is incredible. Dr. Roots. Yeah. የሽጥላ ኢንክሊን ላይ ተፈረቀ ይመሻል። ጎበዝ ናት። ኧረ ደስ ይላል። Jesus. Five years? That's crazy. After 5 years being just Metabolism? That's crazy, man. Because it's not about managing time, it's about managing focus. Yes. የሽጥላ ሩት የሽጥላ ሌላ ሰው ናት። ዶክተር አይደለችም። ሶፍትዌር ኢንጂነር ምናምን ነገር ነው የመጣችው። Senior department. ይችላል ጋይስ ስ ሞዲፋዎችን ምናምን ቀላል አይደለም። ይችላቸዋል አይሰማችሁም። ለምሳሌ በእኛ ባች ኢትስ ፈርስት የማት ተማሪ ነው አይሰማችሁም። ቀላል ለሰው የምትልት እና እንደሌላው ትምህርት ምን አለ የትኛውም የግል የመንግስት ትምህርት ቤት አይሰማም። ከዚህ ይህ ሁሉ ተማሪ መመረቅ ነው። ይህን ያህል ተማሪ ያስመርቃል በአንድ ባች ብዙ ተማሪዎችን ማለት ነው። አሪፍ ነው ወይ ምናምን እየጠበቃችሁ ምናምን ያላቸው አንድ ላይ ያስመረቃቸው የስድስት ዓመት ተማሪ ነው። Imagine yourself as like a lion. Like, if you try to examine that, that's a shame. It's just because I'm not living on the text. No reaction video, no, anything. Crazy.
Hi guys, እንዴት ናችሁ? አሁን ቤት ገብቻለሁ። ቆዩ አክቹዋሊ ትንሽ አረፍ ብዬ አሁን ደግሞ ለማታ መዘጋጀት ጀመርኩ። ማይ ሜካፕ ጠዋት እንደነበረው ነው። ትንሽ ስቲል ላይ ነካ እናደርጋለን። አልለቀቀም ግን አይላሽን ንቅል ነው ያደረኩት። አልቻልኩም ቢካዝ ያ በጣም የሚቆረቁረኝ አልወድም። ዝም ብዬ ማስከራ ነው ለማድረግ ያሰብኩት። ምሳ በላን እዛው። ብዙ አልቀረስኩላችሁም። ከዛ በኋላ ስቶሬጅ ፉል እያለ በጣም እያስቸገረ ነበር ስልኬ። ምሳ በላን ቆየን እስከ 9 ሰዓት። ከዛ ቤት መጣሁ፣ ትንሽ አረፍኩ። አሁን ተነስቼ ልብስ እየቀያየርኩ ነው። ናው ለስ ኮል ማም። ምክንያቱም ይሄም እንደቀድሙ ከኋላ በገመድ ነው የሚዘጋው ኮርሰት ተደርጎ ያ ማይ ሙሉ መዘመያስ ልብስ አገላብጥ ነበር በጣም ማለት ነው። ኤኒዌይስ ዲስ ኢዝ ዘ ድሬስ ሶ ኪዩት። ቅድም ቢጫ ነበርን አሁን ጥቁር ነው። ከዚህኛው ጫማ ጋር ነው ማድረገው ሳይጨልም በደንብ ያሳያችሁ ሁሉንም። ግን አም ሶ ታየርድ እግራችን ወለቀም ታየርድ ፎቶ ስንነሳ ምን ስንል በሂል ጫማችን I don't know how I'm gonna survive ይሄኛው። ለማንኛውም ፍላት ጫማ ይዛለሁ። let's get snatched ከዛ እንወጣለን። ሜካፔንም ነካክተን ወዝወዞኛለሁ። ትንሽ ድራይ እናድርገው።
This is crazy. So from this video, what I've learned is I have to work hard. Let me get so graduate Sark, guys. Machine Chistal. Grilling Chistal, because I'm already Chistal. I have to change. So እንቀጥል ማለት በንግግር አንተ ያሉ ሰዓት ሶሻል ሚዲያ ላይ መስራት ራሱ ኃጢአት ነው። ከአስሩ ኃጢአቶች ውስጥ አንዱ ሶሻል ሚዲያ ላይ ብዙ ሰዓት ማጥፋት። ሰዎች ታይሮስን ቅድሙን ጨርስና ወደ ቪዲዮችን ገብተን ቶሎ ቶሎ ብለን ጨርስ። ሌላ መስሎ ዲግሪ ያለ እሱንም መጨራረስ አለብኝ። የዛሬ 5 ዓመት ኢማጅን ዛሬ 3 ዓመት አንዱን ዲግሪ የሚጨርስ ግላይሰሮል ሜታቦሊክ ባዝ ዘ ሚድፖይንት ኦፍ ግላይሰሮል ኮል ሌላ ጊዜ እናየዋለን። ዋንስ ግላይሰሮል ቢካምስ ጂሲፒ ኢት ሃዝ ቱ ቾይስስ go down glycolysis become pyruvate Krebs cycle or go up glycogens become glucose ራሱ ሊሆን ይችላል። once glycerol becomes GP it has two choices ግን መጀመሪያ glycerol enter the metabolic pathway in the midpoint ነው of glycolysis የግላይኮላይሲስን እንትን glycolysis pathway guys እናቴ ትምህር አልመለስም እግዚአብሔር እኔ ደግሞ ዛሬ ነገ ምናምን ነው ምዝገባው ማለት የሴምስተር ብሬክ እያስተዋወሉ ምናምን ነገር እንትን አድርገውታል። ምዝገባውም ቀን አሳልፎታል። ነገ ነበርና ነገ ነበር ምዝገባው ላቆመው ምናምን እያልኩ እያሰብኩ ነበር። ቢካዝ አሁን ሜድ ስኩል ባቆም አይ ካን ቢ ሪች ኢዝሊ ቢካዝ መማረው ትምህርት አለ። ሜድ ስኩል ከመጀመሪያ በፊት እየተማርኩት የነበረ ማለት ነው። that can help me like to በብዙ መንገድ but I want to be a doctor በቃ አሁን ራሱ እሱን ህርት እያደረኩ ነው በጣም it's okay man guys this is like በጣም አሪፍ ይደርሳል ሁሉ ነገር እግዚአብሔር የድካሜን ፍሬ ሳያበላኝ እንትን አይለኝም። የደከምኩበትን ነገር ሳላጭድ እግዚአብሔር ያሳጭዳል። ላይክ ስደክም አብሮ ከቆመ እግዚአብሔር ያሳጭዳል። ይገባል ያለኝ አሁን የቱ ጋር ነበረ? ስንተኛው ግላይኮላይሲስ ስንተኛው ሪአክሽን ላይ ይገባል ያለው? ግላይኮላይሲስ like the glycolysis and the midpoint of glyceraldehyde 3-phosphate ላይ የሚገባው glyceraldehyde 3-phosphate glyceraldehyde 3-phosphate or GAP glyceraldehyde glycerol glyceraldehyde 3-phosphate with 1,3-biphosphate now this is the glycolysis this is hexose kinase okay ዛሬ እየተወራ ነበር ስለነዚህ ስለ hexose kinase this is enzyme ከዛ ስለ ግሉኮ እየተወራ ነበርና ግላይሰሮል የቱ ጋር ነው የሚገባው እያለኝ ነበረው anyways ገና ፓርት ስንማረው ስለምንማረው ለለፈው okay ስለዚህ let's go man fork in the road once glycerol becomes G3P it has two choices go down glycolysis become pyruvate into Krebs and ATP go up become glucose. Why go up? The liver has plenty of energy from burning fatty acid. It does not need ATP from glycerol. So the brain and the blood cells cannot use glycerol effectively. They need glucose. So therefore, the liver sacrifices the potential ATP from glycerol to build glucose to release into the blood for the brain. So is glucose only used for ATP? This is like glucose is only for ATP. This is my question. Pentose sugar pathway. Glucose is converted into ribose to make so this was my question. So glucose is very important not only for ATP. That was my question. ምክንያቱም ቀደም የጠየቁት ጥያቄ ሎጂካል ነበር። ግላይሰሮል is 3 carbon by the way ግላይሰሮል ባለ ስንት ካርበን ነው? ትራይግላይሰራይድ ባለ 3 እንደሆነ አውቃለሁ። ግላይሰሮል ባለ 3 ነው። ባለ 3 ካርበን ነው አይደለም። ሶ ግላይሰሮል ኢንቱ ፋቲ አሲድ እና ዲሶሺየት ይሆናል። ሶ 3 ግላይሰሮል ግላይሰሮል is 3 colorless odorless okay ሁለት ፋቲ አሲድ ይሄዳል። ሶ እነሱንም ሪቪው ማድረግ አለብኝ ማለት ሶ ኤኒዌይስ ምን ያደርጋል ፔንቶስ ሹገር pathway so pentose sugar pathway and pentose sugar pathway shunt glucose is converted into ribose so glucose is converted into ribose it's okay glucose is converted into ribose okay ribose is one amongst carbon no let me ribose sugar last ribose okay so balance sugar where is the image so this is glucose is this structure guys have no clue but okay just do the work you see this so glucose converts into ribose sugar okay with pentose phosphate pathway so pentose phosphate pathway glucose is converted into ribose to make DNA and RNA because ribose sugar is deoxyribose and ribose sugar this is ribose sugar so OH the first ላይ ከታች ሃይድሮጅን ካለ ሀብታሙ ነው ከስር this is OH ቤታ ነው okay deoxyribose እናሳያለን በዲኦክሲራይቦስ እና በራይቦስ መካከል ያለው ሶስተኛው ካርበን ላይ ነው አይደለም ልዩነት ያለው ሶስተኛው ካርበን ላይ ነው ልዩነት ያለው በሁለቱም መካከል ዲኦክሲራይቦስ ከሆነ ዲ ነው የሚያያችሁ ሰው አይኖርም ማለት ነው this is their difference alright so this is their difference አያችሁ the third carbon atom one oh I thought it was the third one, but the second one, okay? The second one. Because you have to go 1, 2. I actually this one. Deoxyribose. So this one converts into so that it can make with different pathway. For antioxidant defense and fat synthesis. So antioxidant defense is very important. Antioxidants are ፍሪ ራዲካልስ ዲፌንስ ለማድረግ ናድ ፒኤች ኦኬ ትናንትና ስለ ናድ ፒኤች እና ስለ ናድ አይተናል እነዚህ ነገሮች ሴቭ አላደረኳቸውም እንዴት አሁን ፓተርን መቀየር አለበት ሜን I don't care about አሁን ላይ ላይፍን in weeks and months ነው መኖር የጀመርኩት it's not about days because the days are weak the months are strong the weeks are በጣም strong because pattern is very essential በሰዓት አልኖርም በቀን ነው የምኖረው so every action must be follow some because oral exam ሊኖርብኝ ስለሚችል putting this into good note is very very very very wise so ሁለቱ መካከል ያለው ልዩነት what was it rph እና this is electron this one is electron don't acceptor ነው acceptor it is left electron donor no electron okay but this one is used for like different purposes like genesis synthesis and detoxification so you see detoxification detoxification so antioxidant defense no antioxidant free radicals defense so there is an enzyme called it's okay fat synthesis antioxidant defense. Okay, synthesis ብያለሁ. So NADPH is very essential. So glycosylation, attaching sugar to proteins, glycoproteins for cell signaling. So glycosylation, attaching sugars to protein for cell signaling, because out of let us check this. Okay, go ahead. How is glucose produced? Exogenesis. So is glucose directly from food, or is a process which produced በምን ብንጠይቅ exogenesis? So ከ0 እስከ 4 hour after eating food, starch or sucrose or lactose, digestion in the gut absorb glucose directly into the blood. So glycogenesis, stored 4 to 24 hours after eating. Mm, So after eating, stored glycogen in the liver. So the enzyme glycogen phosphorylase chops glucose off the glycogen tree. So glucogenesis. Uh, so this is the internal production you are asking. So ለምሳሌ ከ4 እስከ 24 ሰዓት ባለው ሰዓት store glycogen in the liver, then the enzyme glycogen phosphorylase chops glucose off. እሺ ከዛ ከ24 ሰዓት በኋላ አፍተር ስታርቬሽን ከሆነ ሊቨር 90% ወይም ኪድኒ ኮርቴክስ 10%። ኦኬ ሶ ኢንግሬዲየንት ላክቴት ፍሮም መስል ወርኪንግ ዊዛውት ኦክሲጅን ቢካዝ አይ ኖ ላክቴት በት ላክቴት እንዴት እንደሄደ አላወኩም ነበር። ሶ አሚኖ አሲድ ስፔሲፊክሊ አላኒን ፍሮም ብሬኪንግ ዳውን መስል ፕሮቲንስ ኤንድ ግላይሰሮል ይሆናል የምንጠቀምበታለን ማለት ነው። ፋቲ አሲድ attach of glycerol cannot be turned into glucose. Glycerol fatty acids አይቀየሩም actually they are cannot be turned into glucose. Okay, ወደ ግሉኮስ አይቀየሩም fatty acids. So fatty acids አይቀየሩም ወደ ketone body ነው። Glycerol backbone can be turned into glucose. So where does blood glucose come from after 3 days of starvation? After 3, all dietary glucose and stored glycogen are gone, so the body maintains blood glucose entirely through glycogenesis in the liver. In kidney, the liver synthesis this new glucose using glycerol, lactates from anaerobic metabolism, glucogenic amino acid mainly alanine. While this precursor could be used for ATP, the liver prioritizes converting them to glucose to supply the brain, red blood cells, and which rely on glucose as a fuel source. So how red blood cells use glucose? You are 100% correct. RBC ፔንቶስ አይተናል። ሶ NADPH ሌላ ጊዜ አያቸዋለሁ፣ ግን አሁን I have to be broad. So my technique is this. So my technique is I have to go broad. Okay, የአስተማሪውን እንትን መጠበቅ አይጠበቅብኝም። pace. So there are a ton of videos online and books. So I have to go deep and broad so that I can really play the game. Because አሁን ላይ አስተማሪ ይገባል የሆነ ነገር ያወራል and ወፍ ነው መሆኑ ምንም ብርቅ አይደለም። በቃ እውቀት ነው አይደል? መጽሐፍ ውስጥ ያለ ነገር ነው ማየት ይችላሉ። ሶ እንደዛ ባርቅ ይሻላል። ሶ ፎር ናው ማለት ነው። ሶ ሌትስ ፊኒሽ ቱዴይስ ሪቪው። ለማንኛውም ለክፉም ለደጉም የዛሬው ኢዝ ላይክ አም ብላክ በቃ አለ ብላክድ አውት ብላክ አውት ብላንኮጀነሲስ። ሶ የዛሬውን ሪቪው እናድርግና ዛሬ አሳይመንት ማስገባት አለብኝ የኮምፒውተር ሳይንስ። ጂሰስ ክራይስት። ሶ ፓይሩቬትስ ፎርሙላ here lactic glycerol is polyol sugar alcohol while it is closely related. Okay, so glycerol reacts when the body fat broke down into energy. ጨርስ ለማንኛውም ለክፉም ለደጉም። ሶ km ቫልዩ እና vmax እናየዋለን። ሶ thermodynamics, allosteric site, PKU, structure, so muscle iron pumping አይተናል። ሶ clinical Enzymes serve as biomarkers for identifying organ damage. Okay, cool. Myocardial infarction, heart attack, elevated levels of CK-MB, creatine kinase, yeah, creatine kinase MB in the blood indicates heart muscle damage. Okay, ሊሆን ይችላል. Elevated level. Okay, blood was all right. So, okay, ለነገሩ enzymes have two functional and non-functional. So, if they are functional, they can be found into the blood, in the blood. But if they are non-functional, they are in the tissues. So, creatine kinase, if it is found in the blood, so which means there must be a damage. Liver function test, elevated ALT and AST indicates the liver injury of inflammation. So there are two enzymes as a biomarker for organ damage. Inborn error metabolism, glycogen storage disease, ለምሳሌ deficiency in glucose 6 phosphatase leading to hyperglycemia during fasting. hypoglycemia ከሆነ የግሉኮስ እጥረት ማለት ነው። ሶ ትንሽ ነው ያለው ማለት ነው። ሃይፐር ከሆነ ግን ብዙ አለ ማለት ነው። ሶ ዲስ ሃይፐር ከሆነ ብዙ አለ፣ ሃይፖ ከሆነ ትንሽ አለ እጥረት ማለት ነው። ሌላ ደግሞ ፌናይል ኬቶኑሪያ ዲፊሽንሲ ፌናይሊን ሃይድሮክሲሌዝ ማኔጅድ ባይ ሎው ፌናይሊን ዳይት ኤንድ ታይሮሲን ሰፕሊሜንቴሽን ማኔጅ ሊሆን ይችላል። እሺ እንቀጥል። ሊቨር ላይ ከሆነ ኤልቲ ኤንድ ኤስቲ። ሶ ፋርማኮሎጂክ in importance enzyme as drug target. So how many drug works through enzyme inhibition? Competitive inhibition, the drug compete with the substrate for the active site. Statins inhibits HMG CoA reductase to lower blood cholesterol. Competitive inhibition, the drug compete with the substrate for the active site. Statins, this is statins drug, inhibit HMG CoA reductase. Actually, this is not acetyl CoA to lower blood cholesterol in statins. ስለዚህ ነገር በደንብ ባወራ አሪፍ ነበር። Specifically, like statin drugs for pharmacologic, how this work, because cholesterol is converted by some kind of a process. So let's read about this. Okay, so let me copy paste. No. So here are my notes, and pharmacologic importance of enzyme as a drug. Actually not as a drug, but as a supplement for drug. So for example, statins inhibit HMG CoA reductase to lower blood cholesterol. So I haven't get this point. I don't get this point. Like cholesterol, right? So cholesterol is produced by what? Because here, from my notes, cholesterol statin drugs can be lifestyle drugs. So, like for example, hydrocarbons, you increase glucose, then increase pyruvate, and that pyruvate acetyl CoA, then that converts into cholesterol. This is the pathway. I may be wrong, but this is the process, and it's actually produced in the liver. You can check me, but how does this literally work? So then the competitive inhibition, the drug competes with substrate for the active site. All right, the drug, like there can be a lot of examples for this. So chemotherapy drugs like methotrexate inhibit dihydrofolate reductase to stop cancer cells from replicating DNA. So pathology መጽሐፍ ፓቶሎጂ ቅዳሜ እናነባለን። ፓቶሎጂ every single day. So pathology every single day ቅዳሜ እናነባለን። እሺ እንዳትረሳው ቅዳሜ every single day, because ፓቶሎጂ በጣም ሾክ ያለው ላስት ሰምስተር ከሾኳቸው ትምህርቶች አንዱ ፋርማኮሎጂ ኤንድ ፓቶሎጂ ነው። ሶ ፓቶሎጂ ጀነራል ፓቶሎጂ በጣም ነው የሾኩት። ሶ I will not watch a video, but read a book. I will read a book, especially in immunology and cancer ላይ በደንብ because it's very important man. Like በጣም ነው ሾኩት like እዚህ ጋር አሁን ህመም አለ አይደለም this ልብስ ሳጥብ ምናምን የቆሰለ ነው like I know this is inflammation. This inflammation now I know the science ግን I can't explain it. You have to explain everything. All right, so let's continue. Detoxification, cytochrome P, this enzyme is very essential for phase one. Phase one, so responsible for biotransformation, changing fatty soluble toxins into water soluble form for excretion. ለኤክስክሬሽን ጊዜ የሚጠቅም ድራግ ነው። ለድራግ ሜታቦሊዝም ላይ ይጠቅማል። ድራግ ሜታቦሊዝም ላይ በጣም essential it is for and then there is also cofactors and vitamins. So let's say apoenzyme ማን በጣም እየሸተተኝ ነው እያፈነኝ ነው ማን አየር ላስገባ በጣም ቁጭ ያልኩት ብዙ ሰዓት ጂሰስ ክራይስት አንተም ጂሰስ ጥቁር በጥቁር ከመሆን ብዬ ነበረ። እሺ ሌትስ ጎ ጥቁር በጥቁር ከመሆን ብዬ ነበረ። እሺ ብርሃኑ ከሚያስቸግር ወደዚህ እናድርገው። Oh mama. Okay, so this is apoenzyme. So these are apoenzyme, the protein part. Apoenzyme. So enzyme milk allo. This is literally whole enzyme. So something proteinous እንደሆነ እንረዳለን ማለት ነው from the notes. So apoenzyme and whole enzyme. Because enzyme ማለት cofactor is not the part ያልሆነ መሆኑን እንችላለን እናውቃለን ማለት ነው። So the protein part plus cofactor. So the protein part to ሲደመር the cofactor is whole enzyme. But apoenzyme ባለው only the protein part ነው። So enzyme ትልካለች we know. But the cofactor often derive from water soluble vitamins. ለምሳሌ vitamin B complex that assist the enzyme in catalysis. እሺ እንቀጥል ወደኋላ እንደቀረው እየተሰማኝ ነው something is off because I have to do a lot of problems. So binding site enzyme distinct from active site. When allosteric activator binds to this site, it induces conformational change in the active site, making it suitable for the substrate and switching the enzyme from inactive to inactive form. So hemoglobin transport oxygen from the lungs to peripheral tissue. In tissue, high concentration of hydrogen and CO2 act as negative allosteric effector and inhibitor. So what some people like Jesus Christ, people are very complicated creatures, man. I don't know ከሰዎች ጋር እንዴት ማድረግ እንዳለብን. So thermodynamic catalysis, Jesus Christ, they are very complicated. You don't know them, man. ማወቅ አትችልም ሰዎችን በጣም አንተ ይገርማል በጣም ዳይቨርስ ናቸው ሰዎች የምሬን Jesus Christ, they are very diverse. You can't predict human beings. So anyways, biological example, hemoglobin transport oxygen from lungs to peripheral tissue. So in tissue, high concentration of H plus in CO2 act as negative allosteric effectors or inhibitors. Hydrogen እና CO2 ካለ in tissue, hemoglobin transport oxygen from lungs to every peripheral tissue. We know it. But in tissue, high concentration of hydrogen and calcium, carbon dioxide act as a negative allosteric effector. So allosteric site እናውቀዋለን አይደለም? So allosteric site is nothing but ይላል አስተማሪያችን nothing but so nothing but so allosteric site is nothing but a site which is not the active site ማለት ነው. So this can be this is, let's say this is an active site and this can be an allosteric site and boy boy boy boy boy boy boy boy boy da da da da da da da da da da da da da da da. Here are some cars, man, like 5 years. Don't, Jesus Christ. Why would I care about people in Yemrin? ለነገሩ እኔ ኬር አላደርግም። አይ ኬር ግን በቃ I should stop care. I don't care ለነገሩ I don't care about ብዙ ነገር ብዙ ኬር የሚሰጠኝ ሰው አይደለም ግን they are complicated. When you say በቃ ወደዱኝ ቀረቡኝ ግን they leave you alone. They are complicated. Like peoples are complicated. ስትቀርቧቸው አይደለም. When you become mysterious, they want to know you. When you become open up, they close you just. They read you and now they close you up. That's the mystery. And ከዚህ ሁሉ ዝም ማለት ይሻላል. It's crazy. It's crazy. You can't predict people's mind. እሺ ለማንኛውም this is allosteric site. So hydrogen እና carbon. So hydrogen and CO2, carbon dioxide, act as allosteric site inhibitor. Okay, so allosteric site inhibitor ናቸው ካሉ is it a good thing or a bad thing? Negative allosteric effectors. Okay, negative allosteric. So they bind to allosteric site, change hemoglobin space to inactive for oxygen binding, which triggers to unloading O2. So hemoglobin preferral tissue ላይ ከደረሰ በኋላ ኦክሲጅኑን ማራገፍ አለበት አይደለም እንዳይሆን ያደርጋሉ። ሶ ሃይ ኮንሰንትሬሽን ኦፍ ካርቦን ዳይኦክሳይድ እና ሃይድሮጂን አየን ካለ they may act as negative allosteric effectors and negative allosteric site effectors. Positive ከሆነስ? That's my question. What about positive allosteric site? Okay. What about positive ቢሆንስ ለምሳሌ what about positive ቢሆንስ ለምሳሌ positive አለው like ለምሳሌ so for example allosteric site as enzymes often have multiple subunits quaternary exist two states T tends inactive and are relaxed inactive okay so allosteric site have two states below so tense or relaxed tense or inactive or relaxed or active right. So these are two sites. By the way, there are two states, not sites, but there are two states. It is tense or active. So ቅድም ያልኳችሁ ኔጌቲቭ አክቲቬተርስ ናቸው ብዬ አልነበረም? ሃይድሮጅንና እዚህ ጋር ሃይድሮጅንና CO2 are their negative activators. So what they do is from relaxed state, they change it into tense state. Okay. Yeah, that's what they do. Literally, that's what they do. People are very, very dangerous. You can't predict them, no? What the hell? Jesus Christ. I don't know, but man, like it's okay. It's okay, man. It's okay. It's okay. It's okay, man. It's okay. It's okay, man. It's So now, so if it is inhibitor, shift the equilibrium to T state. So tense ይሆናል. So if it activator, shift the equilibrium to arb state. So this is allosteric's side. So unlike simple enzymes, hyperbolic curve እንዳላቸው ታይፕ ከርቫቸው ላይ ስለ ኤንዛይም አሁን ከተወራ ኖርማል ላይ ለምሳሌ እንደዚህ አይነት ከርቭ ነው የሚኖራቸው። ሶ ከርቮ ምንድን ነው የሚያመለክተው? ዴር ኢዝ ሁለት ሰዎች አሉ። ሶ ዴይ ትራይ ቱ ኳንቲፋይ ዲስ ፕሮሰስ enzymatic process ላይ ማለት ሶ ሌትስ ሴይ ዊ ሃቭ enzyme and substrate, then that convert into can be reversed by the way. So this is if it is 100% or if the concentration of substrate is highly greater than the enzyme is called saturated. So በዚህ ሰዓት before product is reached, there is called enzymatic transition state. Okay, enzymatic transition state, then this convert into what? Product plus what? Enzyme. Okay, because enzyme ይቀየራል substrate ግን ሄደ ማለት ነው። So the substrate converted into buckets. This is the ATC. So let's say this is K1 and this is K2. Okay, you don't know, man, like what the hell? People's love being like የሆነ መጽሐፍ አለ the human nature and people's love being ghosted. That's bad thing አይደለም ግን they love being ghosted. So anyways, let's go back. So ምን እያለን ነው መሰለሽ? So this is the whole process actually, and let me check it out. ቼክ አድርግ ደግሞ ሜን ላይ በጣም ብሮድ አድርጌ ደግሞ ከምጠፋ it's okay. So this is so AS stuff. So enzyme this is the transition state የምንለው ይሄኛው state ነው። Before reaching this, there is a state attain የሚደረግበት. So this is called enzyme substrate complex. So reactants to release the product and regenerate the free enzyme, so they can also go back. So to determine the speed of the matrix and the overall substrate to distribute the rate of reaction, rate of the velocity. So let's create v velocity and along the substrate. So there are good guys that created this formula. So like Vm, concentration of substrate over Km plus substrate. So Km is rate constant. So ይህን ግራፍ ስትሰሩት this is the Vmax. So there is a place called Vmax. So ይህ ነገር አዳድ እያደረጋችሁበት ብትጨምሩ there will be some state maximum velocity that can be attained እና ከዛ በኋላ የፈለጋችሁትን ያህል substrate ብትጨምሩ no more change ማለት ነው። So this is Vmax. So this is the normal function of the normal equation. This is a normal equation of normal workflow of enzyme reaction, enzymatic reaction. But when it comes to allosteric site ላይ ያለው this active site ላይ ያለ ነው። So ለምሳሌ እንደዚህ ሆነ አይደለም። if it is non-competitive ለምሳሌ non-competitive ከሆነ ግራፉ ይቀየራል። ለምሳሌ Vmax ሆነ አክቲቭ ሳይቱ ላይ ለምሳሌ ይሄ ነበራችሁ። Then we created Wakanda. Wakanda, Wakanda, Mukunda, Makanda, Dadi dan dan da dan di dan dan da di dan da, Wakanda, Wakanda. So here Nabara. This is your active site in Belina. In Belina Nyazo, in Belina Nyazo. So this is active site, your active site. Jesus Christ. What did I do to do? And welcome. Bum, bum, bum, bum, bum, bum, bum, bum, bum, bum. What did I say? You're welcome. What can I say except you're welcome? Jesus. So እዚህ ላይ የሚገባው let's say ይህ ቢሆን ከዚህ ጋር ኮምፒት የሚያደርግ ሌላ አይነት ቢኖርና let's say this is the so እንደዚህ ቢሆን so ምንድን ነው የሚሆነው ኮምፒቲቲቭ ኢንሂቢሽን ላይ so this ቅድም ላይ km ቫልዩ ያልኳችሁ the rate ማለት ነው km is just half of the Vmax ነው the Vmax half ሲሆን is just k value km rate of reaction km is like or ደግሞ በሌላ አባባል km ራሱ ሲሰራው ምን ያደርገዋል this is the forward reaction preferable one this spontaneous this is also this is spontaneous no this is spontaneous but this is non-spontaneous ወደ ኋላ መመለስ ማለት ነው this if it is this like a graph so ወደዚህ እንዲሄድ የሚያደርገው አለ k1 and this is the state ከዚህ ወደዚህ transition state then okay so the km value is km value k2 plus k3 over k so km is just the substrate concentration at which Vmax V is just Vmax over V is just Vmax over 2 የሚሆንበት place concentration line this is concentration here this is Kmax so this graph is hyperbolic this hyperbolic not parabolic this hyperbolic hyperbola እንደዚህ አይደለም የሚሰራው then you have this asymptote so like that kind of unreachable asymptote የትም ጋር asymptote ሊኖረው ይችላል እንደዚህ ራሱ ሊሆን ይችላል okay so this is Kmax so now my question is what if there is a competitive inhibitor እዚህ ጋር if it is a competitive inhibitor adding more concentration can improve አይደለም so Vmax does not change for Vmax doesn't change because it is minimum Vmax doesn't change but this may be changed the km can be changing Vmax ሳይቀየር ይቀየራል ማለት ነው so ግራፍ አሪፍ ግራፍ ካገኙ ልፈልግ competitive and non-competitive graph. Yeah. There in the internet, okay. አሪፍ አግኝቼዋለሁ። አሪፍ ኢሜጅ አግኝቻለሁ። ኮምፒቲቲቭ በሚሆንበት ጊዜ the V maximum is not going to change, but you need more substrate. Okay, that's ልዩነቱ። Yeah, this is the graph. What would I say except you're welcome? This is free enzyme. ይኸው እንደምታዩት then for competitive inhibitor this is the K is changing concentration half ከመድረሱ በፊት ያለውን ይህን ያህል ነው ሄደው እዚህ ጋር K ይኸው እዚህ ጋር ነበረ ለምሳሌ 5 ሊሆን ይችላል እዚህ ጋር ግን እዚህ ጋር ደረሰች ማለት ነው half ለመድረስ so Km is always የVmax over half ነው ኦኬ half ከሆነ የደረሰበትን ፖይንት so non competitive ከሆነ ግን already Vmax ይቀየራል but Kmax will not change. Okay, so የቅድሙ ጋር ሳይደርስ look at this or ደግሞ የVmax half so ግን በእንትኑ ግራፍ ደግሞ በእንሲሆን ደግሞ በደንብ ነው የሚያሳይ ፒውር እና ኮምፒቲቲቭ ኢንሂቢተርስ ላይ በሊኒየር ዌብ ወርክ ኢኩዌሽን በሚሆንበት ጊዜ በአሪፍ ማየት ይቻላል። ሁለት ሰዓት ያህል ነው ያወራሁት። ምራቄ እየመጣ ነው። ግን አሎስትሪክ ሳይት ሲሆን ሲግሞይድ ግራፍ ነው የሚሆነው። ዲስ ኢዝ ፓራቦሊክ አይደለም። ለአሎስትሪክ ሳይት ግን ዲስ ካይንድ ኦፍ ነው የሚሆነው። ሌትስ ሴ ዲስ ኢዝ ቪማክስ ላይክ አይነኬው እንደዚህ ይሆናል። ግን ሞር ላይክ እንደዚህ አይነት ነገር ነው የሚሆነው። ዊች ሚንስ እንደዚህ አይነት ሲግሞይዳል ነው የሚሆነው። ችን ግራፉን ነው Jesus Christ, people are very different. You can't predict people. Jesus Christ. Good lesson. እችኛዋ በደንብ ኖት መያዝ አለበት። ፈተና ላይ ሊመጣ ይችላል፣ ኢንተርቪው ላይ ሊመጣ ይችላል። ለኢንተርቪው አይደለም የሚዘጋጀው፣ ለደንበኛ ለፔሸንት ነው የሚዘጋጀው። አንድ ሰው ፈትነኝ ፈትነኝ እሱ አይደለም ዋናው ነገር ማወቅ አለብኝ። አሎስትሪክ ኢንዛይም እግዚአብሔር ደግሞ ፍሬ እንድበላ ያደርገኛል። እግዚአብሔር ዘርቼ ዝም ብሎ አይበትነውም። አይ ነው በቃ እግዚአብሔር መልካም ነው። እግዚአብሔር ያደርሰኛል። መልካም አምላክ ነው። አሎስትሪክ ኢንዛይም ካይኔቲክስ ቅድም እንዳልኳቸው አሎስትሪክ ሁለት አይነት ስቴት ብለናል። አር ማለት ሪላክስድ ስቴት፣ ቲ ደግሞ ቴንስ ስቴት ነው። ቴንስ ስቴት ላይ ሎ አክቲቪቲ ነው። ኢፍ ኢት ኢዝ ኢንሂቢተር ከአር ወደ እንደዚህ ነው የሚቀየረው። እዚህ ጋር አሁን ኢንሂቢተር ኢዝ ዲስ ሉክ አት ዲስ ዲስ ኢዝ አክቲቬተሩ ወደ ከዚህ ወደዚህ ይቀይረዋል። ቬሎሲቲ አየው ኤስ አይነት ሼፕ ነው። ዲስ ኢዝ ኖርማል ሪፍሌክት ቦዝ አር ኤንድ ቴስትድ ሊዲንግ ቱ ሲግሞይድ ከርቭ ኦኬ ዘን activators push toward R state. Activators push toward R state. This is R state እንደሚያሳየ and this is also low state. አየው this is R state just two state ብለናል this is T state so uh, what they do is so activators they will push it toward R state and so kind of S kind of shape okay this is one of the difference because for example glycolysis PFK1 glycolysis it is inhibited by high ATP and activated by this PFK1 glycolysis ላይ ማለት so በኋላ ላይ እንትኑን እናያለን reaction እናያለን this is not the main concern of the course but this is the examples these are the examples እግዚአብሔር ይረዳኛል እግዚአብሔር ይረዳኛል እግዚአብሔር ይረዳኛል የምር እግዚአብሔር ይረዳኛል I trust in God እግዚአብሔር ይረዳኛል አልፈራም በቃ ለምንድን ነው የምፈራው ህይወትን የሰጠኝ እግዚአብሔር ነው ለምን እፈራለሁ so ጂ ጋውት ወይም ሪያ የሚባል በሽታ አለ ተብሏል እግር የሚያነክሱ ሰዎች አሉ አይደለም so አልኮል እና ስጋ ምናምን ጥሬ ስጋ አትብሉ እሺ ለምን እንደሆነ ታውቃላችሁ so alcohol increases free radical production and is rich in purines so አሁን ፒውሪንን ላስረዳችሁ ነው ማለት ነው purines are Purines are a group of organic compounds that play a vital role in biochemistry. They are nitrogenous base that derivates the purine structure. So, group of biochemicals nature. And let me show you their example. ምክንያቱ የመጨረሻ ሰማይና ምድር this is ለቅድሙ maybe purines እኮ ነው man what the hell so like እዚህ ጋር ሆነ ዘር purines ነው እኮ purines አረላሽ በጣም አንተ ጉግል ግን አንተ በጣም አሪፍ እየሆነች ነው ጉግል አሪፍ እየሆነች ሰርችን እያስታወሰ ነው ላይክ እንዴ አረላሽ በጣም አሳድገውታል what the hell what the hell ነው የሚባለው what the hell actually አይደለም በክርስቶስ ያገኘሁት ነፃነት እግዚአብሔር ምሕረት ጸጋ ምሕረት የዘላለም አምላክ ቸርነት የማያልቅ ቤተ ክርስቲያን purines አዶኒን እና ጓኒን አይደለም purines okay so ክትወረን ሳውን ታትማን በልቤ purines are like double length ናቸው pyrimidine ጥሬ ስጋ ፒውሪን አለው lot of questions so ምንድነው ያሰብነው መሰላችሁ ልጆች ክላስ ላይ ጥያቄ ነው የምሰራው ጥያቄ እሄዳለሁ፣ ፕሪንት አደርጋቸዋለሁ። አስተማሪው ሲለፈልፍ ጥያቄ ይሰራሉ። 3 ሰዓት ምናምን ያህል የማይገባኝን ነገር አለፈልፍ ስሰማ እንትን አለ አላባክም ጊዜ። እስካሁን የተማርኳቸውን ምንድን ነው ማድረገው ክላስ ላይ ዲስ አወር ፕላን ሰው ክላስ በምገባበት ሰዓት የተማርኩትን ጥያቄን ፕሪንት አድርጌ ወስዳለሁ። ኖቴን ምናምን ነገር እንትን አድርጌ እስከ መግባት በፊት ግቢ ውስጥ ፕሪንት አስደርጋለሁ። በቃ ፕሪንት አስደርጋለሁ። በቃ እንደዛ ነው ማድረገው። ካሁን በኋላ አሪፍ 100 ምናምን 30 60 200 ጥያቄ በቃ እጨርሳለሁ። እነሱ ምንም ማድረግ አልችልም። እስካሁን የተማርኩት ላይ ጥያቄ ይደቀደቃል። ፋይናል ሚድ ኤግዛሞቹ ፕሪንት ይደረግ ሚድ ከዚህ በፊት ተማሪዎች የተፈተኗቸው ሚድ ኤግዛምስ አጋዥ መጽሐፎች ጀርባ ያሉ ለምሳሌ ባዮኬሚስትሪ ዳውን ማርክስ የዩኤስኤምኤልኤ ወር ደግሞ አቦማስ ምናምን ላይ ያሉ ጥያቄዎችን ሰርች እናደርጋለን። ኤቭሪ ክላስ ስገባ አስተማሪ ያልከተ አስተማሪው አሪፍ ካልሆነ ብዙ የሚቀባጥር ከሆነ አዲስ ማይገባኝ ነገር ከማድረግ አንድ ወደፊት አንድ ወደኋላ አንድ ወደፊት አንድ ወደኋላ ነው። ኦኬ ቀጥል የዛሬውን ሪቪው እናደርግና ምንድን ነው እዚህ ጋር አሁን እያደረግን ያለነው አስፕሪን አክታስ ሪቨርሰብል። ስለዚህ ኢን ፒውሪንስ ዲስ ኢንክሪዝ ዩሪክ አሲድ ፕሮዳክሽን ዩሪክ አሲድ ፕሮዳክሽን ይጨምራል ብለዋል። አያችሁት እንደምታዩት እዚህ ጋር ዩሪክ አሲድ ከሶዲየም ከናይትሮጅን ጋር ከአሞኒያ ጋር ምናምን ግንኙነት አለው። ከናይትሮጅን ዳይኦክሳይድ ጋር ከናይትሮጅን ኦክሳይድ ጋር ግንኙነት አለው እንዴ? ሌላው ደግሞ አስፕሪን አክታስ አን ሪቨርሰብል ኢንሂቢተር ፎር ሳይክሎ ኦክሲጅኔስ ኮአክስ 1 እና ኮአክስ 2 ባይ ኢንሂቢቲንግ ኮአክስ 1 it reduces prostaglandins that protect the stomach leading to protein ulcer but also reduce platelet aggregation helping prevent blood clot. blood clotting ይከላከላል ማለት blood clot. reversible inhibitor by cyclooxygenase by inhibiting coax 1 and reduce prostaglandins that protect the stomach leading to potential ulcers but also produces platelet aggregation helping prevent blood clots. Alright, so this is great idea. Great idea. አረዳሽ ለሁለት ሰዓት ያልኩት ብያለሁ። ምግብ መብላት ነበረብኝ ማን ብሬክ አድርግና ደግሞ በላና እግዚአብሔር ግን ይረዳኛል። ዳስ አይ ትረስት ኢን ጋድ እግዚአብሔር አምናለሁ። እግዚአብሔር አይጥለኝም። ዳስ ዋን ቲንግ። ነን ኮምፒቲቲቭ ላይ ቪማክስ ዲክሪዝ ያደርጋል። KM unchanged. The inhibitor does not interfere with substrate's ability to bind to the active site. ለምን እንደሆነ ልንገር። ነን-ኮምፒቲቲቭ ከሆነ ሊትራሊ ኢንሂቢት ቱ አን ኤሌክትሪክ ሳይት ነው የሚሆነው ብዙ ጊዜ ሳይት አደር ዳን አክቲቭ ሳይት ላይ ማለት ነው። ከኮንሰንትሬሽን ጋር ግንኙነት አላት? ምንም የላትም። ኮንሰንትሬሽን ጨመር ካልጨመር ጉዳዩ አይደለም። በቃ ምን አገባው? ጉዳዩ አይደለም፣ አይመለከቱም። ምክንያቱም ያለው ሌላ ቦታ ነው። ሼፑን ነው ቼንጅ የሚያደርገው። ቪማክሱ ዲክሪዝ ይደረጋል። ቪማክስ the inhibitor reduce the catalytic efficiency of the enzyme regardless of substrate concentration. ኦኬ በቃ ግልጽ ነው። ግን K is M is unchanged በቃ እንዴት ነው የምትቀይረው? አይመጣውም። The inhibitor does not interfere with the substrate's ability to bind to the active site. does not በቃ ከሰብስትሬቱ ጋር አልገለም። unchanged ትሏል። ነን ከሆነ unchanged KM በት ኮምፒቲቲቭ ከሆንሽ ግን ለምሳሌ ኤግዛምፕል ልስጥ። ሰልፋ እነዚህን ነገሮች እነዚህን ኖቶችን ልስጥ፣ ኮፒ ላድርጋቸውና እናውራባቸው። ያልገቡ ነገሮች ሲኖሩ ደግሞ ይጠይቃል። ይህን የዛሬውን ሌክቸር ከጨረስኩ በኋላ ምን አደርጋለሁ? ዛሬ የተማርነውን ፓወርፖይንት አታች አደርጋለሁ። ፓወርፖይንቱንም እናየዋለን ማለት ነው። ሽንት ቤት ድረስ እና ልምጣ። ሃቭ አ ናይስ ታይም። እስከዛ ድረስ ግን እነዚህ ናቸው። እግዚአብሔር ግን ይረዳኛል። እኔ እግዚአብሔርን አምነዋለሁ። እግዚአብሔር ግን እንደ ሰው አይደለም። የምር ሰዎች ግን ይለዋወጣሉ። በጣም በጣም ነው የሚለዋወጡት። በምንም ተአምር ላውቃቸው አልቻልኩም ሰዎችን። እግዚአብሔር ሆይ፣ ተቃራኒ መሆን ለምንድን ነው የሚፈልጉት? ቀረብኳቸው ስትል ለመራቅ፣ ስትርቃቸው ለመቅረብ መከራ። ችግር የለም። አይ ትረስት ኢን ጋድ። ሜቢ ሁሉም ሰዎች አይደሉም፣ በእርግጥ ሁሉም አይደሉም። ዜር አር ላቭሊ ፒፕል፣ ዜር አር በጣም ስዊት የሆኑ ፒፕልስ አሉ፣ በጣም መልካም የሆኑ ሰዎች። ኢትስ ኦኬ። ክፉ ሰዎች ስለገጠሙኝ ችግር የለም። ኢትስ ኦኬ። እግዚአብሔር መልካም ነው። በተቻለ መጠን የራስን ፍልስፍና አለመቀየር ነው። ሰው ያለውን ነው የሚሰጠን። ሰው ያለውን ነው የሚሰጠን። ንቀት የሚንቅ ሰው አንተ መናቅ እኮ በረከት ነው። መናቅ ኢዝ በረከት። ሌትስ ቶክ አባውት ዲስ። አንዳንዴ እኔ እንደ ሀሳብ እወዳለሁ፣ ፈላስፋ መሆን ስለምፈልግ ትላለች ስክል ነው። ግን መናቅ ኢዝ በረከት። ባይዘ ማትናቁ ከሆነ በቃ መናቅ በረከት ነው። ሰዎች ሲንቋቸው በጣም በረከት ነው። የሚናቅ ሰው በረከት አለው። እግዚአብሔር ይቆምለታል አብሮ ይቆም። ምክንያቱም እግዚአብሔር ሰውን ሁሉ ፈጠረ አይደለም በምሳሌው ምናምን። ከዛ ኦሎፈሰደ ከተፈጠሩት አፈር መካከል አረ እኔ በልዩ አፈር ነው የተሰሩት ምናምን ስፔሻል ነኝ ምናምን ብሎ የሚያስበውን ሰው እግዚአብሔር ያዋርደዋል። የሚታበይ ሰው እግዚአብሔር ያዋርደዋል። መመካት ራሱ ትዕቢት፣ ባለው ነገር መታበይ ምናምን ራሱ ማለት ነው የመጀመሪያው ኃጢአት እንደሆነ ነገር የመጀመሪያው ኃጢአት መታበይ፣ ፕራውድፉል ሀርት፣ መታበይ ኃጢአት ነው። ሰውን የሚንቅ ሌላውን የሚንቅ ሰው ደግሞ የታበየ ሰው ነው። እግዚአብሔር ያዋርደዋል። የተናቀውን ግን እግዚአብሔር ከፍ ያደርገዋል። ማንም ሰው ቢንቀኝ እኔ ይገደኛል? አይገደኝም። ምክንያቱም ዝም ብሎ ጨምሮልኝ አልጨምርልኝም። ምንም ማድረግ አይቻልም። ምንም ልናደርግ አንችልም። በቃ እኛ ምንም ልናደርግ ስላልቻለን ይቀጥላል። በቃ እኔ ምንም ላደርግ አልቻልኩም፣ እቀጥላለሁ። ምን አደርጋለሁ? ምንም። እሺ ዛሬውን አታች አደርግላችኋለሁ። ዛሬ የተማርነው ሌክቸር ኖትስ እንዳለ እዛ ላይ አታች አደርጋለሁ። እንትን ደግሞ እንደ ቻሌንጅ ዛሬ የማነበውን ከዛ ኦርጋናይዝ አደርጋለሁ፣ ኖሽን ላይ ያስገባሉ። ኖሽኑንም አስቀምጥላችኋለሁ። የዛሬውን ኖት አስተማሪ ያስተማረውን ጭምር ጊታቤ ላይ ያስቀምጥላችኋለሁ። እስኪ ጌታ ፑሽ ይደረግ። ያ ጊታብ ላይ ነው አስቀምጠው፣ ገብታችሁ ደውደራሉ። ጊታብ ላይ አስቀምጠዋለሁ። ጊታብ ላይ አክቹዋሊ የኮምፒውተር ሳይንስ የኤአይ ምናምን እንዲሁም ደግሞ የሜዲካል ትምህርት እንትኖችን እዛ ላይ ስለማስቀምጥ የዛሬ የተማርኩትን ፑሽ አደረግና እንዘጋዋለን ማለት ነው። እስከ ቻልን ድረስ እንቀጥላለን። አሁን ትንሽ ለስቴክ ብሬክ እናደርግና እንመለስ። ሽንት ቤት ልሄድ ብቻ ግን እችን እንጨርሳት። ኬኤም ኢንክሪዝ አፓረንትሊ። አሁን ይሄኛው ኮምፒቲቲቭ ኢንሂቢተር ሲሆን mechanism the inhibitor is a structural analog of the substrate and competes for the same active site. Km increases apparent affinity for the substrate. በነገራችን ላይ ሎወር ኬኤም አለ ማለት ሃይ አፊኒቲ ያለው ማለት ነው። ሎወር ኬኤም ሼር አላደረኩትም እንዴ? if you have lower km which means higher affinity አለ ማለት ነው። that very great thing. It is very very very good. It is very essential. lower km አለ ማለት you have what? If you have lower km you have higher affinity ማለት ነው, and it's preferable. Okay, but km increases apparent affinity for the substrate decrease because the inhibitor is blocking the site. km increase ያደርጋል ማለት ነው። because lower ከነበረ you have higher affinity ነው የነበረበት። አሁን for the substrate ያለውን decrease ስለሚያደርገው km increase ያደርጋል። Vmax ግን unchanged ነው። If you add enough substrate it will outcompete the inhibitor and reach maximum velocity. For example, melanionate inhibits succinate dehydrogenase TCA cycle ላይ አልደረስኩበትም። TCA cycle ግን I promise you አሁን ፌብሩዋሪ ላይ አይደል ማርች ላይ ግን I will understand every bit of TCA cycle. I promise you I will digest it. በደንብ ነው እንትን ማድረገው። We will read books, we will watch videos, we will use every single resource we have to decipher this. I'm telling you I may be slower, ብዙ ላይ ገባኝ ይችላል ግን ሳይገባኝ ግን አላልፍም። I will ask teachers and ተማሪዎችን ጠይቃለሁ። I will search even whatever method ተጠቅሜ እረዳዋለሁ። ሚታዮሌት ላይ እያሳየ ነው። ሶ ኢንሂቢትስ ሱሰኔት ዲሃይድሮጂኔስ ቲሲኤ ሳይክል ላይ ቲሲኤ ሳይክል ላይ አንድ አለ። መያዝ አለብኝ። ሰልፋድራክስ ለልጅ አይሰጥም ቢካዝ ፎሊክ አንቲባዮቲክ ዛት ሚሚክ ፓባ to inhibit bacterial folic acid synthesis ዲኤንኤ ሲንተሲስ ላይ ቢካዝ ዲኤንኤ ሲንተሲስ ሳይደረግ ወደ ሴል ሳይክል ስለማይገባ this is very important. I don't know what I'm talking about, but I have general overview clue. So statins ደግሞ inhibits CoA reductase to lower cholesterol. ቅድም ስለ ኮሌስትሮል አውርተናል። እንመለስ three type of reverse inhibition. So there are three type of ለምሳሌ reversible and irreversible inhibition ላይ enzyme inhibition ላይ inhibitors are molecules. Okay, question 10 ልሽናል። You see this? Yeah. The sound يقال الصوت ኦልራይት ሶ በልተን መተናል። ምግብ ምናምን እየበላው ነበር እና ትንሽ ብሬክ እናድርግ ምናምን ብለን ነበር። ሶ ሌትስ ጌት ባክ ቱ አወር ስተዲ ሌትስ ፊኒሽ ቱዴይስ ክላስ ኦኬ። Let's get okay. All right, so why is my the cholesterol inhibits feedback inhibition ለምሳሌ the final product of metabolic pathway acts as allosteric inhibitor for the first or rate limiting enzyme in that pathway. This prevent the wasteful overaccumulation of products. ለምሳሌ ሄማ inhibit ALA synthase and cholesterol inhibit reductase palmic እነዚህ ነገሮች feedback inhibition So feedback inhibition, product inhibition mechanism, the final product of metabolic pathway acts as an allosteric inhibitor for the first or rate limiting enzymes in that pathway. Okay, the final product. So heme inhibitor inhibits ALA synthase, cholesterol inhibits this reductase, palmic acid inhibits, okay, acetylchoic carboxylase, cool, okay. All right, let's go. Okay, suicide case inhibition. Okay. All right. Come. Okay. Okay, suicidal inhibition and suicidal inhibition, a definition, a form of irreversible inhibition where enzymes commit suicide. So mechanism, the enzyme treats the inhibitor as a substrate, converting it into a highly active reactive intermediate that then binds permanently to the active site, killing the enzyme. So suicide inhibition is a form of irreversible inhibition where the enzyme commits suicide. Suicide ማለት ራስን ማጥፋት ነው። So the enzyme ራሱን ያጠፋል ነው። So the enzyme treats the inhibitor as the substrate, converting it into highly reactive intermediate that then binds permanently to active sites, killing the enzyme. What the hell? So the enzyme treats the inhibitor as a substrate. Okay, there is inhibitor and the enzyme. Okay, so this is the enzyme, this is the inhibitor. All right, so this is okay. All right, so this is the inhibitor, this is the enzyme. So the enzyme, but there is also the green one is, let's make it the enzyme here like this, because this is the substrate. Okay. Okay, this is the substrate. So this enzyme acts as, this is the inhibitor. So this enzyme treats the inhibitor as a substrate, which means treating them as this. So what it does is converting it into highly reactive intermediate that binds permanently to active site. So it converted make this inhibitor, convert it and transform it into an intermediary substance, and then so that it can active here, permanently binds to this active site, then killing the enzyme. So that is what it is what it is. Biochemistry, it is what it is. አሁን ይህን ስታዩ it's just bonds, it's proteins folding, nothing else. ህይወት ያለው ኮንሽስ የሆነ ነገር የለም እዚህ ውስጥ እንደ ባክቴሪያ እንኳን አይደሉም but they know, they interact. This is crazy. So allopurinol, allopurinol used for gout. Xanthine oxidase converts allopurinol to alloxanthine, which then reversibly inhibits the enzyme. So this allopurinol used for gout, then xanthine oxidase converts to allopurinol to alloxanthine. Okay, so great. ماشي ሌላ non competitive inhibitor. No more. Okay, so non-competitive inhibitor. Non-competitive inhibitor. So the mechanism, the inhibitor binds to an allosteric site. So the inhibitor binds to an allosteric site. So a site other than active site. This is non-competitive inhibitor. So it can bind to free enzyme. Then so let's move on to this. Heavy metals, lead, mercury binds to sulfhydryl group. So non-competitive, uncompetitive inhibitor እና non-competitive የተለያየ ናቸው። ምክንያቱም the inhibitor binds only to the enzyme substrate complex አቺ ነገር ናት በቃ ለያቸው። This dude is the ES complex bind የሚያደርገው inhibit የሚያደርገው so that product አይመጣም ነው። This usually happen in reaction involving multiple substrates. ለምሳሌ ካይነቲክስ ቦዝ km and v decrease እዚህ ጋር ሁለቱም ናቸው። ሶ እዚህኛው ነን ኮምፒቲቲቭ ላይ ከሆነ ማን ዲክሪዝ ያደርጋል? ቪ ማክስ ዲክሪዝ ያደርጋል። ቢካዝ ኬ ደግሞ ምንም አላረጋችሁ ኢፌክት ስለማይኖረው ይኸው km አንቼንጅ ቪ ማክስ ዲክሪዝ። እዚህኛው ላይ ደግሞ ፊድባክ ኢንሂቢሽን ይሄኛው እነዚህ ፋይናል እንትን ሲያደርግ ነው። በዚህ ጋር ኮምፒቲቲቭ ኢንሂቢተር ላይ ግን ቪ ማክስ አንቼንጅድ ነው። km ግን ኢንክሪዝ ያደርጋል because more substrate በመጨመር ነው ቪማክሱ ጋር አቴን ማድረግ የምትችሉት። Okay, these are type of reversible inhibition, but also he discussed about the type of enzymes. This is by the way type of enzymes አውርቷል። Okay, so type of enzymes, for example, so enzymes used for primary mechanism to facilitate reactions. So these are mechanism catalytic strategy actually. So እነዚህ እናያቸውና God is so good. God is so good, የምር God is so good. Jesus, never man, never. Doesn't matter, it doesn't matter. Everything will change. ሁሉ ነገር ይቀየራል። ፐርማነንት የሆነ ነገር የለም። My current situation ጨምሮ nothing is permanent. Everything will change. Everything will change. Everything ይቀየራል። Nothing is permanent. Everything will change. So acid-base catalysis. So rather than that. So acid catalyst involves transfer of proton. Styrene is the crucial amino acid here because SPK is closer to physiological pH, and this is used as a buffer. እንደ በፈር ምናምን እንጠቀምበታለን። የስታዲንን ፕሮፐርቲ እናያለን። We use it as a buffer. Okay, we use it as a buffer. Styrene is used as a buffer. Styrene is amino acid with polar charge side. So positive የሆነ side charge ነው ያለው. So it acts as a buffer, act as a both proton donor and acceptor. Okay. Covalent catalysis. Temporary covalent bond is formed between the enzyme, often using serine and cysteine. Cysteine have, by the way, SH bond አላቸው. So cysteine is special case. SH bond አለው. So it can create disulfide bonds. ሁለት cysteines አጠገብ ላጠገብ ቢሆኑ they can create disulfide bonds. Let me show you this thing. ሰልፋይድ ቦንድ መፍጠር የሚችለው አንዱ እሱ ነው። ሲስቲን እንደ serine. You see this cysteine? Okay, so temporary covalent bond is formed between enzymes, often using serine and the substrate. So catalysis by proximity, mostly proximity ላይ እንትን የሚያደርጉት they are ligases, specifically ligase enzymes. They join, bring to substrate and close together. So ligase ስንተኛው ናቸው? አምስተኛው ናቸው። Ligase are the fifth ናቸው አይደል? Ligase are the sixth enzyme ናቸው. Catalyst by bond strain. Enzyme specifically lyses induce physical strain on the substrate bond, making them easier to break without needing water. So lysis is the fifth one. So these are four mechanisms. So acid catalase, covalent catalyst, covalent bond is formed between enzymes, and the substrate በሁለቱ መካከል then catalyst by proximity. So enzymes specifically ligases bring two substrates close together in the correct orientation to react. Then catalysis by bond strain. Enzyme specifically lyses induce physical strain on substrate bond, making them easier to break without needing. So these are the four mechanisms. So the high overview ነገር ነው ያለው. So let's talk about class of chemical composition hollow enzyme. Simple enzyme composed exclusively of protein nature. Hollow enzyme functional units consisting of protein part, apoenzyme, and a non-protein part, cofactors. Cofactors are divided into metal activated, loosely bound CL4 amylase, metalloenzymes, permanently bound metal ions, coenzyme, loosely bound, often vitamin derivatives, prostatic groups, tightly or covalently bound. Example, hemoglobin, heme, coenzymes, co-substrate, loosely bound, often vitamin derivatives. እነዚህን ያዙልን። ሜታሎ ኤንዛይም ፐርማንትሊ ቅድም ፌሮታኒ ከኮፐር ጋር ፌሮታኒ እንዴት ነበረ ግን የአይረን ስም ይዞ እንዴት ነው ከእንትን ጋር ይሰራል? ኮኤንዛይም this is apoenzyme is just the protein part. Apo holoenzyme is the functional unit constant. So coenzyme so co-substrates usually vitamins often. So vital እነዚህን ላሳይ. There are clinically significant vitamins here. Vitamins and the clinical significance. Many coenzymes are derived. So many coenzymes are derived from water-soluble vitamins. So many coenzymes are derived from water-soluble vitamins. Deficiency can lead to specific metabolic failure. It can lead to thiamine required for decarboxylation. Decarboxylation pyruvate to acetyl-CoA. Deficiency in alcohol can cause lactic acidosis and mental impairment. So lactic acidosis and mental impairment due to interrupted ATP productions. So required for decarboxylation, pyruvate to acetyl-CoA. Deficiency in alcohols can cause lactic acidosis and mental impairment due to interrupted ATP production. And biotin required for carboxylation. It follows the ABC rule. Required ATP, biotin, and carbon dioxide converting pyruvate to oxaloacetate. Biotin convert pyruvate to oxaloacetate. Ascorbic acid, vitamin C, essential for collagen synthesis. It facilitates the hydroxylation of proline. Without it, collagen fibers are weak and leading to bone and skin tissue. So vitamin C is essential for collagen synthesis, and it facilitates the hydroxylation of proline. Without it, collagen fibers are weak. So proline is an exception, by the way. Proline is also kind of ring structure now. Then it's cyclic amino acid now, but hydrophobic side chain yellow ring like cyclic now. So fibers are weak, leading to bone and skin. So cholamine and folic acid crucial for methyl group and one carbon transferase. So one carbon transfers folic acid, one carbon transfer, and crucial for methyl group. ቃል ብቻ ሳይሆን በተግባርም ወሳኝ ነገር ነው። እሱን ጨረስኩ። thermodynamics activation energy as enzymes speed up reaction by lowering required for the reaction the required required to reach transition state line. Analogy catalyst is compared to lowering a high wall. Without an enzyme, the wall is high and few molecules can jump over it. Enzymes do not change the overall energy of the reaction. Allosteric regulation. Allosteric site, a binding site of an enzyme distinct from the active site, where when an allosteric activator binds to this site, it induces conformational change in the active site, making it suitable for substrate and switching the enzyme from inactive end to active form. All right. So biological example, hemoglobin. Hemoglobin transports oxygen from the lung to peripheral tissue. In tissue, high concentration of hydrogen and CO2 act as negative allosteric effector. They bind to allosteric sites, changing hemoglobin state to inactive. ኦኬ ቅድም አይተናል ይህን ፓርት ኦኬ ስለ እንትን አላየንም ክሊኒካል ፒኪዩ ኤልኤስ አይተናል ኦኬ ስለዚህ ተርሸሪ ሳይድ እንደሆነ አይተናል ሶ ስለ እንትን ስለ ክላስ የስ ስለ ክላሶቹ ሶ seven major classification of nomenclature ቅድም ደግሞ ስለ እንትን አይቶ ነበረ ስለ እሱ አውርቶ ነበር አላውቀውም ስለ ኦኬ ችግር የለም ቪዲዮ አይበታለሁ በደንብ ዲቴል እገባበታለሁ ችግር የለም በደንብ ቪዲዮ አያለሁ አነብበታለሁ ኦኬ ማንም ተአምር በቃ ሰውየው አይደለም የሚፈጠረው ይህን ሳይንሱ ያለ ነው በቃ ተጠንቶ የተቀመጠ ነው ማወቅ ይችላሉ ማንበብ ይችላሉ I'm ready to learn I'm ready to learn to read አላውቅም አውቃለሁ በኋላ it's okay trivial name often short and commonly used example hexokinase okay trivial names like hexokinase systemic name more descriptive detailing the substrate and reaction type example ATP and ATP is what እዚህ ጋር ATP is acting as an inhibitor አይደለም እዚህ ጋር and this hexo I don't think ATP is as an inhibitor okay ቆይ እንትኑን ፓወርፖይንቱን እናየው ፓወርፖይንቱን እናየው የሰውየውን ፓወርፖይንት በደንብ ማየት አለብን sorry to kind of yeah. Oh shit. Oh shit. እሺ ሶ እንመለስ። 2018 ላይ ኦኬ ዲስ ኢዝ ዘ ዲጂት የመጀመሪያው ክላስ ነበር። ክላስ ኤንዛይም ቤዝድ ኦን ዘ ሪአክሽን ታይፕ ሰብ ክላስ then subclass and specific serial number. This is very very important. Okay, so የመጀመሪያው is oxidoreductase facilitate electron and hydrogen transfer. Example dehydrogenase enzyme. So today on my reduction and oxidation reaction ነው በደንብ ቀጥታ ማየው ቪዲዮ because I forgot this because high school የነበርኩት 2013 ምናምን high school የነበርኩት እኔ 2013 አሁን 2014 የጨረስኩ 2013 ባች ነኝ anyways. So I was good student chemistry ላይ high school ያለ transferase move chemical groups amynyl acyl phosphate between molecules. so move የሚያደርግ ከሆነ phosphate so ATP ላይ የምንጠቀመው እንትን ይሄ ነው ማለት ነው። ATP ADP transferase ናቸው ማለት ነው። ATPase enzyme ATPase ማለት ፕሮቶን አይደለም so I think that's oxidoreductase I guess. So transferase Hydrolysis break chemical bonds using water. So all digestive enzymes hydrolyze lysis enzyme lysis enzyme yeah I will like in the zone like so lysis enzyme cleave bond without use of water. Without the use of water is cleave bonds without the use of water. Okay, you don't use water. Fuck it. You don't use water. Okay, without the use of water. Isomerase, rearranging atoms within a molecule to change its structure. Example, turning glucose into fructose. Ligase, join two molecules together, often require ATP, DNA ligase. Translocase, facilitate the movement of molecule or ion across the biological membranes. So translocase is the new one ማለት ነው። ሶ ምን ያደርጋል? ፋሲሊቴት the movement of molecule or ion across biological membranes። ኦኬ ዲፕ መነበብ አለበት ስለዚህ ጉዳይ። ለጊዜው ግን ሊግላጌዝ join two molecules together often requiring ATP ሶ DNA ligase ሶ ያበሰልኩት እየደረሰ ይመስለኛል። every notes and ኖቱን እንትን እናደርጋለን። So ligase isomerase rearrange atoms within a molecule to change its structure. Example, turning glucose into fructose. Isomerase enzyme isomerase class ነው። So ligase ቅድም ምን ብለናል? ligase በፕሮክሲሚቲ ብለናል። ላይሴስ ደግሞ restraining ብለናል። ላይሴስ cleave bonds. Hydrolase breakdown for digestive enzymes. Typically digestive enzymes using water ነው። without water ያልነው የቱ ነው? Without the use of water lysis. Reductase redox transferase move. Okay, so all right, so is it right. List of questions. Shift አድርጎ ስታለንስ ፋርማክስ Factors affecting enzyme activity. So substrate enzyme concentrate increase concentration generally increases velocity until saturation point is reached. So temperature human enzyme have optimum temperature approximately generally increases velocity until a saturation point is reached. Temperature human enzyme have an optimum temperature approximately 37 degree. Activity drops significantly beyond 40 degree due to protein denaturation. So each enzyme has an optimum pH. Pepsin at pH 2 intestinal enzyme at pH 7-8 deviations alter the ionization of active site, hindering substrate binding. So activator and inhibitor specific ions like chloride for amylase can activate enzyme. Conversely, oxidants can inactivate enzymes by damaging sulfhydryl groups as the active site. And UV radiation can cause structural damage denaturation. So each enzyme has an optimum pH. Okay, pepsin, pH አላች temperature. How pH change ከተባል deviation alter the ionization of active sites hindering substrate binding ብለህ ትመልሳለህ። ቴምፕሬቸር ከተባለ activity drops significantly beyond like denaturation ሊሆን ይችላል enzyme have optimization and due to protein denaturation substrate concentration increase concentration generally increase velocity until saturation ግን አሁን ለምሳሌ ነን ኮምፒቲቲቭ ላይ ከሆነ አይሆንም cannot change velocity አይጨምርም but decrease ነው የሚያደርገው okay because extra autosomal activism amylase conversely oxidants የሆነ ነገር አስታውሼ ነበር የተረሳሁት ኤቲፒ ምናምን ጋር ማለት ነው ኤቲፒ used as something like example ሰጥቶ ነበር ሰውዬው inborn error metabolism almost ጨርሰናል አሁን የያዝኩትን ኖት ጨርሼዋለሁ የያዝኳቸውን ኖቶች ጨርሻለሁ let's go over የሰውየው ፓወርፖይንት ላይ because I hate reading PowerPoint by the way አላቅም ለምን አላቅም ያቀነጭራሉ ምናምን የሚባለው በጣም ቀላል ነው እንትን የሚያደርጉት ኖት ለአስተማሪ እንዲመቸው አድርገው የሚያወጣው እና ላይገባ ይችላል አብዛኛው ጊዜ let's try እና let's give it to the manga እሺ ሶ አታች የማደርገው እነዚህ ነው እሺ በኋላ ላይ አታች አደርጋቸዋለሁ ስጨርስ አታች አደርገዋለሁ እነዚህን አንድ ላይ ይህን ፓወርፖይንት ከዛ ሌላውን እነዚህ ዛሬ ነው እዚህ አካባቢ ነው ያየሁት ይችላሉ እነዚህ እስካሁን የወሰድኩት ያወራው ሲጋር ኢንዱስድ ፊት ሞዴል ምናምን አምጥቷል ሲምፕል ኮንጁጌት ፕሮቲንስ ሶ አርኤንኤ ፖሊመሬዝ cell RNA is made up of protein groups and non-protein components. Example, RNA polymerase conjugate enzyme the side chain of the common amino acids provide only a limited range of chemical reactivity and carrier properties. The side chain of a common amino acids provide only a limited range of chemical reactivity and carrier properties. So, enzyme for the last semester line. One of my weakness was reading the PowerPoint because PowerPoint I hated PowerPoint so ሳላነብ ነበር ብዙ ጊዜ እና ብዙ የተበላሁት ፓወርፖይንት ባለማንበብ ብዙ ሰው ይህን ብቻ በማንበብ እስኪ አሁን ላንብበው ኢሰንሻል አይንስ እነዚህን አላነበብኩም ኢሰንሻል አይን አላነበብኩም so activator ion and metal activator enzyme ions necessarily but loosely and reversibly bound often participate in binding of substrate. Metalloenzyme ions tightly bound and frequently participate in catalytic reactions, so particularly in active center transfer electron bridge the enzyme and substrate stabilize enzyme conformation and neutralize the ions. Okay. እኔ የፈለኩት ይሄን አልነበረም ብቻ እንያቸው እዚህ ጋር ያሉት these are what essential ions ማለት ነው so transfer electron bridge the enzyme and substrate stabilize the enzyme conformation neutralize the ions so ferroxidase copper carbonic anhydrase zinc cytochrome oxidase copper and iron China ferroxidase cofactor so the enzyme ferroxidase the cofactor is zinc carbonic anhydrase carbonic and zinc ነው ሳይቶክሮም ላይ ነበር ያየሁት cytochrome oxidase. Cytochrome four acids. Okay, so copper in iron hexokinase. Hexokinase is like the first one again. ግሉኮስን ወደ ቀይረው ሄክሶካይነስ ማግኔዥየም ያ ከኤቲፒ ጋር ማግኔዥየም አለ ማለት ነው። ሴሌኒየም ግሉታዮን peroxidase selenium and arginase manganese. Okay, the xanthine oxidase molybdenum. Okay, now coenzyme organic compound small and chemical stable compound transferring electron protein and other metabolic molecules. Okay, co-substrates correspond to enzymes able to be separated with dialysis and dialysis accepting H or group when leaving to transfer it into others and vice versa. Proteolytic group tightly bind through either covalent or non-covalent. Okay, transfer co-substrates. Okay. Remain bound to the enzyme during the course of reaction. Jesus Christ told coenzyme metabolite coenzymes they are synthesized from the common metabolites, several in TP and ATP, most abundant UDP glucose. So it's okay. ስንት ገብቶ ይሆናል? Okay. Vitamin driven coenzyme. They are derivatives of vitamins and can only be obtained from nutrients.